FEL project. Are you there?
I am. Can you hear me okay?
Wonderful. I believe so, yeah. Do you want to go ahead and share your slides?
I will, yes. And let's see. Is that coming through okay?
Yeah, if you could just maximize. Oh, there you go. Excellent. Yeah, it's maximize.
Okay, great.
Yeah.
Great. Hey, thinking about the Mpemba effect, I'm not familiar with that, but I would say that's typical of the type of problem that's attacked with an XFEL. As Robert very nicely showed, what XFELs bring to the game is the time dynamics. So not just static structure, which we've had, for a century or more, but then seeing actually how molecules function and how systems evolve over time. That's what they're just excellent at. And so looking at, kind of all the way down to attosecond out to nanosecond dynamics, you can start to make movies of atomic structure, which is very interesting. Okay, I'm going to tell you about the Compact XFEL or CXFEL project. This is centered at Arizona State University. It includes about 80 people from 16 different institutions. And it really grew out of something called the BioXFEL Science and Technology Center, which has started about 10 years ago. Henry Chapman was very involved in that, and our local researchers, Petra Froma and John Spence, and it's been a real leader in particular in bio applications of XFEL and serial femtosecond crystallography. So this ability to take very fast snapshots of chemical reactions as they occur. Things like photosynthesis and the folding of proteins are typical examples. But XFELs do many different things, as Robert showed, and it's broadly applicable. The key thing with XFELs is that they look at time-resolved science, and so that we add, very importantly, function and time dynamics to the atomic structure. This effort has grown. This 2013 was before I was at Arizona State. I came in about 2015 with the concept for a very compact XFEL. This is something that fits in a room. I'll show you in just a moment. And it has grown. It's a two-phase project, I should say. In the first phase, let me get my pointer here. There it is. In the first phase, this is showing our laboratory spaces. This is about 1,200 square meters, the space that you're looking at. So this is in the laboratory of a building on campus, something called the Biodesign Institute. And we have built the first phase already, called the Compact X-ray Light Source, or CXLS, in this area. That's a small device based on a linear accelerator and a laser. So instead of producing X-rays in a magnetic undulator that might be 100 meters long, we use a laser, and it has the same effect on the electron beam under the right conditions, where you can make very short bursts of intense X-rays with a much smaller machine. Because the laser period is much shorter than an undulator period, the accelerator can be much smaller. So our entire accelerator, the entire vault that contains the accelerator here, is 10 meters long. So it's just really a different scale of device. So that's the first phase. We've built that. We're commissioning it now. We've got the electrons and the lasers functioning, and we expect to be looking for X-rays actually not next week is a big holiday in the US, but the week after. So the first week of December, we're hoping to make our first X-rays with that. And that's primarily a hard X-ray device. At the same time, we have proposed to build a second device in this complementary set of laboratories. That's what we call the CXFEL. So the device over here is not yet producing coherent X-rays. It's coherent in the sense that a synchrotron is coherent, but the pulses are short. And the device we propose over here is a soft X-ray device that would be fully coherent. It has full laser properties and can do all kinds of different laser tricks with those X-rays. Okay, and I'm mostly going to focus on the CXFEL. The CXLS, I'll show you a photo in just a moment. This is what we've built already. So this is the accelerator here. The electrons start at this point. They travel, the actual accelerator itself is only about 1 meter long. And then there are magnets and different beamline elements. We collide with the high power laser at this point, and then X-rays are produced in the X-ray hutch over here. And we have an end station like you might find at a synchrotron or another XFEL. And here, this is the laser in the adjacent room. This is what we've built already. This is all operational, and we're just about to begin to look for X-rays. And then the CXFEL we proposed actually in 2021 and are awaiting a decision from the National Science Foundation. We hope it goes forward as soon as February of next year. We expect to begin construction at that time. And so one thing I should point out here is that, and again, these are engineering images, so these are all to scale. The accelerator for the CXFEL is virtually identical to the accelerator that we've already built. These are fairly small devices, but you see that the laser is growing in size, and we need a much more intense laser when we're making the XFEL. And so, working with our partners at Trump Scientific in Munich, we're developing this very high power laser for that purpose. These experiments, the XFELs are a type of laser, and more, I think people are recognizing that XFELs and the facilities around them are fundamentally laser facilities as an evolution away from accelerator-based facilities. This is showing the CXLS as it exists today. On the left here in the yellow device, that's a solenoid magnet. That's where the electrons are produced. Robert mentioned, and there was a question about the picosecond laser. We actually use a femtosecond laser and then photo emission at the cathode to produce the electrons. This point here is just about 2 meters away from the cathode, and that's the end of the accelerator there. And then the electrons travel down this beamline and collide with the very high power laser here. The X-rays are produced and travel through the far wall and into the X-ray hutch. This is now operational. In a couple of hours, I'll be over there running beam again today. And so here's a full layout of the CXFEL. This over near, this is where the electrons begin. They travel in this direction, produce X-rays here, go through a thick wall not shown, and then into the X-ray hutch where there are end stations. And there are more lasers. This is the very high power laser that creates the X-rays. And then there are pump lasers. Typically, experiments at XFELs are done as pump probe experiments where we will photo excite a particular reaction that we're interested in studying and then track the evolution of the sample with the X-rays. So these pump lasers initiate those reactions, and they span a very wide wavelength range from ultraviolet to terahertz, actually. And to give you an idea of the scale of this, the CXLS that we have built already, that's about a $20 million investment in that. The CXFEL, I'll show you our budget in a little more detail. This is our proposed budget to the National Science Foundation. It's got a baseline cost of just over $70 million, and a little more than half of that is equipment, and then about $30 million in labor. And we have a proposed 5-year construction schedule for that. Oh, I should mention too, this particular device has a single end station at a time, but we can move different end stations. These are all different end stations that we can move into place. We have precision mounts that we can quickly move one or another in. So if we're switching from biological experiments over to, say, atomic molecular optical experiments, it needs a different apparatus, a different sample delivery system, and we have self-contained devices that switch them in quickly. So in that cost estimate I show you, there are 5 distinct end stations included. This is, I'm going to go through a little bit of the technical detail of the accelerator and the laser. I'm just going to skip very quickly. It would be a long talk to go through all that. And then I'll describe the science that we hope to do. So this is showing the accelerator. The beam starts down at this end. These copper-colored structures are the copper linac. In the CXFEL, we don't use so-called SASE, self-amplification of spontaneous emission, to create the X-rays. that has statistical fluctuations, as Robert showed very nicely, in the pulse length and in the bandwidth and in the spectrum that's emitted, in the energy per pulse. We do something different. We actually diffract the electrons where my cursor is, and then we take that diffracted electron pattern, accelerate it through the accelerator, and do an accelerator physics trick that switches the diffraction pattern from the spatial dimension into the temporal dimension. And by doing that, we can produce fully coherent X-rays when those microscopic electron pulses collide with the laser. And so way back here where we diffract the electrons, we can, of course, create many different patterns depending on what we use to diffract them through. And that can change, for instance, the color of the X-rays that are produced. It can change the pulse length. We can produce multiple pulses. We could produce a chirped pulse, each pulse exactly like the one before it, so greatly reducing those fluctuations. This small device has certain advantages and disadvantages over the large devices. It's much smaller, first of all, so that means it costs less, but it's also much less powerful. The flux out of this is about 10 to the 8th photons per shot. That's our predicted number. So that's about 10,000 times less than the big machine. So it's less intense. However, the photons are all in a single coherent wave packet, and we have full control over the properties of that packet, over the phase, the amplitude, the color that it makes. And so it has very interesting applications for science. Okay, and then so after this admittance exchange, we collide with the laser and produce the X-rays, and they go onto the hutch. And then in this slide, the whole device is shown again, but I want to focus in this area on this very high power laser that's being developed by our partners at Trump Scientific. So it's a 600 millijoule, 1 kilohertz laser. It's producing picosecond pulses nanosecond pulses out of the amplifier. Those are then compressed into the picosecond range. And then they go into this very large device called a Harriet cell, which has a low gas pressure in it. And through nonlinear generation of phase fronts, there's a broadening of the spectrum in there. And we're able to compress that picosecond pulse down to about 40 femtoseconds. We lose a little bit of energy, but the output of that is expected to be roughly 400 millijoules in a 40 femtosecond pulse. This is an extremely powerful laser pulse. And that's what we then collide with the electron beam to produce the X-rays. So this is a first of its kind type of laser. It's a first of its kind type of accelerator too, using that electron diffraction and admittance exchange. So that's some of the technical detail: laser, accelerator, and now the end stations. There are 5 different end stations. This one, the XIS, is a crystallography station that actually goes in the CXLS, and so that's hard X-rays in this kind of 10 to 20 keV range. And then the rest of these are primarily looking at spectroscopy and time-resolved experiments in the soft X-ray range, in the range from about 300 eV up to about 2.5 keV up to the sulfur edge. And so there's a base end station here that can take different modules depending on which group is using the X-rays. And we expect in about 2 or 3 days to be able to swap end stations, and then to have a run typically of 1 week to 2 weeks for each experiment. The experiments are primarily in 3 different areas. Of course, continuing the work of the BioXFEL group, there's a lot of biological sciences work. Usually it's looking at time dynamics. So we're looking at structural electronic oxidation changes, for instance, in photosynthesis, or looking at catalysis, looking at catalytic materials in biology and how the catalytic cycle proceeds. So we have long had X-rays, synchrotrons to look at static structure, but now we're starting to look at the dynamics. What are the chemical reactions? How do they proceed? How do we optimize these? How do we, increase the efficiency of photovoltaic materials, say, or really understand what's happening inside of proteins and viruses? In AMO sciences, we're looking at conical intersections and also, again, photoactive materials, for instance, interfaces between different materials. How are electrons exchanged? How is energy exchanged between materials? What are the time scales? I should mention that the CXFEL is naturally a very short pulse device. A long pulse for us is about 10 femtoseconds. Those are the longest pulses, and the shortest are about 300 attoseconds. It is designed for extremely short pulses. And then the third major area is in quantum materials research. So this is materials exhibiting novel quantum behavior, such as correlated electron devices, motions, high temperature superconductors, or in these unusual arrangements of spins and magnetic vortices and things. So these are often ephemeral structures. The physics that's being looked at happens on very fast timescales, and you need a very fast probe in order to take these kind of stop motion movies of them in action. So looking at biological materials, at AMO physics, at quantum materials research, and then pulling that all together, Abbas Ormaz, our collaborator from the University of Wisconsin at Milwaukee, is applying artificial intelligence, machine learning to the data that's collected. As Robert pointed out, you often take hundreds of thousands or even millions of shots. This particular device operates at 1 kilohertz at 1,000 shots per second. So in not very long, you can accumulate 1 million shots. And there's some variation, some timing jitter, some jitter in the spectrum that comes out. And using AI and machine learning, you can greatly reduce the apparent timing of that. So this is showing an LCLS result where an experiment that had a physical timing of about 100 femtoseconds, using software techniques and sorting of the images, able to reduce the unknown jitter to just a few femtosecond scale. And so AI and machine learning with these kind of statistical variation and large data sets can have a big impact. So that those techniques can be applied across all of the different sciences. I think there's always interest in budget. So this is our projected budget for the new machine, the CXFEL. These are the total costs of the different areas. And you can see, generally an XFEL is $1 billion project or €1 billion project. And most of that is tied up in the very long accelerator. But here the accelerator is shrunk quite a bit, and so is a much lower cost device. And the costs are spread among the lasers, the science end stations, and other facilities that are needed. Significant laboratory facilities are needed for any of these cutting edge devices. So our split is about 40 million in equipment and 30 million in labor for the total project cost. And it's a 5-year construction project. It would take us about 5 years to build this. Okay, these are the key performance parameters of the device. These are what we want to meet, the so-called threshold, or the minimum that we can do science with. And then the objective parameters, this is what we actually plan to meet with our device. And so showing you certain parameters, you can see that the pulse energy is in the range of nanojoules to perhaps 100 nanojoules. And that's coming in extremely short pulses. And to put our performance kind of into the larger context, this is a chart of peak brilliance now. And so the big XFELs are up here, Flash and the Euro XFEL and LCLS are in the 10 to the 33 range. Our CXFEL is in the 10 to the 27 to 10 to the 28th. This is brightness in standard units. So you can see that we're off in our own little unoccupied space here. And the synchrotrons are very bright on an average basis, but less so on a peak brightness, which is for the single shot types of experiments that we're doing. That's the key parameter. Laser is also very bright, but of course, longer wavelength or lower photon energy they occupy out there. Okay, and as I mentioned, we're not based on spontaneous emission, and so we have great control over the properties of the X-rays that are produced with this device. And with that, I'll take questions.
Thank you very much for the nice talk, Bill. Sure. Any questions? Please go ahead. while I'm waiting to see hands, I actually have a question myself. So I'm just trying to see. I don't see any hands yet. Oh, actually, there is a hand. Yeah, where is it gone? Oh, Tabitha again. Yeah, please go on.
Hi, Bill. Very nice talk. You showed us the cost of construction, but maybe it's because I'm on the small screen. What would be the operational cost, continuous operation, after the construction period is over? Thank you.
Sure. Yeah. So the question is, what's the operational cost? We think it's about $8 million per year. That's what our estimate is. And that's roughly 10% of the construction cost. That's a good guidance for any of these capital intensive projects, typically about 10% of the construction cost.
Can I just add to that question as well? You mentioned that the brightness is a few orders of magnitude below. And of course, the cost is a few orders of magnitude below a large scale facility, such as the European XFEL. How about the energy demands and power requirements? How do they compare?
It's orders of magnitude below. We're at, I know for our operating machine, which is very similar to the one we'll build, we're at about 100 kilowatts average power. That's wall plug power. It's less power than the air conditioning in the building, for instance. So the upgrade machine, I would say it's under 200 kilowatts average power.
Okay,
and the big ones are multi-megawatt. I don't quite even know what they are. Probably 10 megawatts or so. Okay,
so it's conceivable that there could be more of these facilities built. This is the only CXFEL, I think, in the world at the moment operating. Is that right?
Well, it's not operational yet, but
almost operating.
We are pursuing. I know of other groups are pursuing. This basic idea of colliding electrons and lasers to make X-rays there are great advances in both technologies, in the accelerator technologies and especially in the laser technologies. And I think it's inevitable that this will build over time. It is, I should have pointed out, you're absolutely right. We wanted to bring the XFEL properties onto campus, which is why we're building this, but we very much want to see this technology spread throughout the world, to Africa, to other countries, to other institutions. It's the kind of thing where, a particular medical institution or an industrial plant could have their own. And so it's a bit of a different model. We definitely welcome users from around the world to come and do experiments in our machine. But the whole idea here is that you can have one at home, that you don't have to necessarily go to a faraway place, but can build your own.
Lawrence?
Yes, so we have at the African Light Source Foundation, we have actually been looking at this model of compact light sources, several of them in Africa campus based, because for all the reasons that you would do it, because you can do research duty cycles, and then you can also do class duty cycles. You could imagine the material science course having labs where you actually load a sample of something.
Right. Yeah, we have about 25 students in the laboratories. So it is, being university based, it's a natural, and that's in the construction of the things. It's a natural for the students.
And so the Dutch Smart Light project they're operational, is that correct?
I think they're not quite on yet. But they're getting close. Jan Leuten, who I know well, is leading that from the technical perspective, the accelerator perspective. And I'll find out more. We've actually just joined together. We've started a so-called network of networks. It's an NSF term, where we join with other facilities. It's been primarily now in Europe and the US so far. We're actually just initiating this effort where we take these kind of what we call mid-scale facilities. So this is, something like what we're building is bigger than what a single professor might have in their laboratory, but it's not as big as the very large machines. We call them mid-scale. And we have our own, sets of challenges and advantages, and we're trying to team up with others that are interested in the same kinds of technologies and science, and join together to build things. We've done
Right,
that in the US with, we plan to do complementary experiments with other groups in the US. Ohio State and the University of Michigan are our partners in this. And we all have these mid-scale facilities that we want to share users and kind of coordinate schedules and that kind of thing to make it more useful to the broad community.
That's right. And so I'm familiar with this mid-scale facilities. Tess is a good friend of mine. And we're actually, the African Light Source Foundation is trying to plug into Tess's program and also to Linda Horton and her team to look at this mid-scale facilities solution for Africa. So
that's great.
Well, I'll be in touch with you about that. And I put into the chat an old DOE report about compact light sources. I actually did want to ask about other methods of creating the light, like plasma acceleration, and have those other technologies sort of died, or is ICS kind of the way that's happening?
Let's not offend people, or at least let me offend them. The plasma accelerator is very interesting. You know, I'm an accelerator physicist, and they are fascinating from a source design point of view. What they really are, though, are a way to replace a large accelerator with a much shorter accelerator, but still making a high energy beam. And the high energy, so what we've done is gotten rid of the need for a high energy electron beam. So our top energy is a few tens of MeV versus the GeV, thousands of MeV that the big facilities have. And that's because we use the laser to collide with. So the laser plasma device, its great advantage is its strong accelerating gradient. So it can get to a high electron beam energy in a relatively short distance, but then you still have the long undulator and all of the downstream equipment that would be associated with that. So my personal approach is that if we're only at tens of, I have a conventional accelerator and it's 1 meter long. I think you saw in the pictures that the laser is actually bigger than the accelerator. So I'm not trying to shrink the accelerator anymore. I think we need to shrink our lasers essentially. But plasma Wakefield and laser plasma accelerators, they're very interesting devices. It's an interesting way for future applications of the large devices, I would say. Okay.
Well, let's try to reprise that DOE workshop that was now 12 years ago.
Yeah.
So we'll be in touch on that.
Recall it. Sure. Yeah, I hope it would be great if DOE, likes to build the large facilities. They're not always crazy about the small ones, but it would be great if there's enthusiasm for that. We have a lot of our collaborators are from the DOE labs. SLAC and Lawrence Berkeley Lab in particular are helping a great deal with the design, both on the accelerator side and the end stations and X-ray experiments.
Marcus, if I can make one more comment, if there's time.
Okay, very quickly, go on. Yeah.
So about the comment about replacing the high energy beams. So Lincian had a customer that was convinced that if they had a machine like a compact light source in their laboratory on their campus, that they would actually be more users of like Diamond because they could refine their techniques and create more samples. And so,
right, we don't think the small ones are going to replace the big ones. They're different. I like to compare us to a scalpel. We're a particular tool, and the big ones might be a hammer or a power tool. And they can do science we can't do. I think with our coherent control, we'll be able to do science they can't do. Mm-hmm. You could take one of these small ones actually and seed the large one and transfer those coherent properties to the large one and stabilize the output would be one great application of the small ones. So I think they're very complementary, and I think you're right. We can grow the user community overall. It's as people know, it's very difficult to get beam time at the large facilities. There are 5 in the world, soon 6, And it's tough. They're expensive and hard to access. And if we could have many more where people could develop their techniques, I think it's very complementary.
Very nice. Just one more question from the chat. So could you please comment on the application of the CXFEL in medicine?
Yes. We have two devices here. We have the CXFEL, which is the coherent laser. And in terms of medicine, I think its largest impact is likely to be in the development of new drugs, in understanding how drugs work and really having insights not just into the molecular structure, but into the time dynamics of how they function. The pharmaceutical industry, of course, has a large presence at synchrotrons and I would say a growing presence at XFELs. But beyond that, the CXLS, which is partially coherent, not fully coherent like the laser, but the CXLS is a very interesting device for medical imaging, because now you can begin to do coherent imaging at the synchrotrons. Very nice experiments have done in medical imaging using phase contrast and other kind of partially coherent techniques that are not transferable to a hospital because you can't put a large synchrotron into a hospital. And we do think there are applications not only in medicine and medical imaging, but also in museums and looking at cultural artifacts for the other device, the so-called incoherent device.
Very nice. Well, thank you again. I think we're going to have to thank our speaker in the interest of time and move on. So thank you again, Bill. Very nice talk. I'm sure there are many more people with questions who can probably follow up later on.
Okay, yeah, happy to talk.
Wonderful. Okay, so we'll move on to our next speaker. We're going to switch to Henry Chapman this time round now. So Henry's going to speak. Hopefully you're still there. Yes, I can see you. And Henry Chapman, of course, from DAISY, and he will talk about serial femtosecond crystallography. So we're looking forward to your talk. Thank you again, Henry.
Okay, yeah, thanks, Marcus, and thanks also to Gideon, who I guess was scheduled to give a talk in this at this time. We had a little mix up with time zones and times, but so I'm glad you could all help me out here. And yeah, because I'm really happy to talk about serial femtosecond crystallography, which is a method that sort of began at the very first, when the first hard X-ray FEL started operations. That was the linear coherent light source in California. And Robert mentioned this, yeah, so that was in 2009. So I'll just sort of begin though by stepping back quite a bit further, earlier, and to talk about diffraction. Here's a very sort of iconic diffraction pattern that many people know. It's the diffraction of DNA, double-stranded DNA, which was made by Rosalind Franklin. This is not of a crystal, but of just fibers of DNA that were kind of bundled together and put in the X-ray beam. And so what's interesting here is that that the structure is really encoded in this diffraction pattern out to sort of atomic resolution, and from sort of this encoding, in this case, well, Rosalind herself understood that this was showing that the structure was helical, but Crick and Watson sort of also got a peek at this and realized that it was actually a double helix from a missing layer line that's here due to a sort of interference effect. And we know that if looking at things you might have done at in the lab or at school, that's if you just shine light through structures, you can learn a lot about them. You can basically, if you shine light through a slit or diffract from a hair, you can sort of figure out how thick the hair is by looking at, say, a laser pointer just scattering off that. If you have two hairs or two slits, you get interference between them, showing up in this modulation. So you get destructive and constructive interference. 3 slits gives you quite a different appearance. And really the scale that sets this distance is the wavelength of the light, right? So if these slits are brought together really close, then the fringes get broader and broader, and eventually you can't see them, distinguish them as two slits anymore when they're sort of overlapping to within a wavelength. And this, of course, is why we want to use X-rays for exploring matter at the kind of atomic scale, because the wavelength of X-rays is the interatomic distance. So you can sort of figure out structure here, but it requires more to sort of go in the inverse direction from a pattern like this to figure out exactly what the structure is. Although for slits it's easy, you can sort of just say, okay, I can figure out the distance between them. But essentially it is an imaging technique. If we did have a nice X-ray lens that could give us atomic resolution, of course we'd use it, and we'd have beautiful X-ray microscopes that could image molecules. And if we did have that, what we would do is just put that in that diffracting beam. The lens essentially recombines that wave to form the image. If we sort of decompose this process into particular spatial frequencies of the object, because we can represent any object as its sum of frequencies. I mean, that's the operation of a Fourier transform, just a mathematical representation. And if we consider one of those spatial frequencies, or sort of it and its negative self, it will diffract into these 2 orders. And what the lens does is bring those orders together, re-interferes them to produce a fringe pattern. So that's good, but it does it to all frequencies and interferes them all coherently so that they reform the entire image of the object. Now, unfortunately, we don't have that lens for X-rays, so we are forced to just measure the diffraction at this plane instead of using a lens. And now this particular spatial frequency is measured in the form of the intensity at a particular direction. So we've got this angular resolved detector, for example, or it could be film, but usually we use pixel array detectors. And so whatever's sort of at this pixel is telling you the strength of this particular frequency. And so we can see our frequency content of the object. But what we're missing is really how to recombine these. And in particular, that's encoded in the phase of the diffracted wave. And essentially that's telling you, when you recombine all these frequencies, it's telling you where to put that fringe pattern, or the lens in this case knows where to put the fringe pattern. But if we shifted this pattern or this fringe, if we shift this backwards and forwards, the phases in these beams changes, and that's not. But the intensity is not, and it's only the intensity that we record. So we're insensitive to the shift, and so we don't know how to basically place all these frequencies to form the image. And this is a process of sort of phase retrieval, and which has had a long history of how to solve it. And perhaps the first sort of real application of trying to sort of really beat that phase problem was in crystallography. And here, going back to the late 1950s, there was a decade of effort to try and figure out how to do that phasing. It was found that the proteins could crystallize when you grow big enough crystals and put them in the X-ray beam. Your diffraction pattern changes quite considerably compared to, say, that fiber diffraction. You now get Bragg spots. It's sort of the inverse of the slits that I showed, which give fringes. Now you're sort of having a periodic system, gives you sort of sharp diffraction orders in reciprocal space. But it's the same thing. It's essentially encoding the structure of the constituents of the crystal, the molecular constituents. Now, Max Roots figured out a great way of doing this, which was to measure a crystal and then in this case of hemoglobin, and then soak it in mercury and have mercury atoms bind in specific locations and measure again. And by looking at the difference, you could sort of see where these very heavy, strongly scattering centers are, and sort of use those kind of like a holographic reference to figure out the phases. So crystallography has had a long and really storied history, and even a lot of involvement in Africa. And sort of in this whole period, which kind of matches the period shown by Robert in this Moore's law growth of the sources themselves. So over the time that Robert was educated and became married and had kids and whatever and got a job, there was a lot of development in crystallography and in protein crystallography. And you see this large rise, which is represented, or is the number of entries in the protein data bank, which is a worldwide repository of structures that are solved by various methods. And actually the blue here are the X-ray structures. There's NMR as well, and electron microscopy, which is actually growing rapidly through cryo-electron microscopy. But you see that it's not just the numbers; it's also all these nice, beautiful structures giving insights into life, into structural, into biology, how it works, how it functions, and solving diseases or working against diseases and things like that. But of course, most of these structures are indeed static, and it's this development here was really spurred, as I said, by the development of sources. So synchrotron radiation sources, having facilities with undulators and beamlines and software and detectors and all this sort of thing. But there was always an issue with protein crystallography, which has been the problem of radiation damage, which we've already talked about a bit during the last session. So X-rays are energetic, of course, necessarily because we want the short wavelengths. So of course they have quite a bit of energy per photon. They're ionizing. They can knock out electrons, photoionize, and then there's a whole, even after a photoionization, you get the photoelectron coming out and an Auger electron, which goes on and hits other atoms and ionizes them. And so you can get a whole cascade of damage to your sample as that energy is distributed throughout the sample. And it's, unfortunately, there's more photoabsorption events than coherent scattering events, which lead to the information in your diffraction pattern. So the amount of damage per sort of scattered photon is quite severe. And so the only way to sort of overcome that was to make a big enough crystal that you could expose it with a low enough exposure that you didn't exceed too much dose into your sample. This is illustrated here by this picture of a virus crystal taken at Diamond Light Source by Robert Owen and Elspeth Garman, and showing the real catastrophic effect of just exposing it for half a second at a very sort of modest, very low exposure. This is done at room temperature, but this is quite a sensitive crystal, and you can see this if you expose it again here, you get you can get diffraction here for a while until it degrades. Yeah, so as I said, the only way to overcome this was to make a larger crystal that would give more scattered photons for a given exposure and really spread out that energy across more mass, essentially. Now, there's another way around that, which became sort of feasible with the development of these femtosecond X-ray sources, which is to essentially freeze out all the damage and all the motion using short pulses, which is nicely illustrated in this old photograph by Harold Edgerton, who from MIT was one of the first guys to develop flash photography. So you see that the inertia of this top part of the card has left it there, despite the damage. And that inertia of atoms is what can really save us in avoiding the damage and increasing the exposure many thousands of times onto a sample. And this was kind of really pointed out very graphically here in this paper by János Hajdú at University of Uppsala and Richard Neutzer, who was working with him at the time, and these other people. They just did molecular dynamic simulations and asked what would happen if you suddenly ionize with a very short pulse every single atom in your sample. And of course, it blows itself apart because of that Coulomb repulsion, but it takes some time for the atoms to get moving. And you've got around 10 femtoseconds of time to get that clear picture before the destruction has occurred. So these pulses from the FELs, and this is a plot that Robert showed, really had the properties required to do this. And it was such a huge jump from what was available before, 9 or 10 orders of magnitude higher peak brightness from the fact that we get many more photons per pulse and in much shorter time. So as I said, at that time, when FELs first came online, it was still an open question whether this idea would really work, and could you outrun radiation damage to the atomic scale. And so we thought, well, to investigate this, I mean, we'd been doing earlier experiments at the Flash free electron laser in Hamburg, which was a much softer energy, so long wavelengths. We couldn't access the atomic scale. So we thought what we'll do is put in a crystal into the beam coming out of this very long undulator where the Sussy process has occurred, and just kind of see what happens. And we were expecting with the pulses that we were told we would get, which were about 100 femtoseconds, so that it would be useless. And so we'd need very big unit cell crystals to see, maybe a few Bragg peaks that would indicate just some remnants of an exploded crystal. But sort of to our great surprise and relief and good fortune, we really saw Bragg diffraction out to very high resolution, out to the highest that we could measure, and this was even with these very delicate crystals of photosystem 1, which were provided by Petra Frommer, who was really part of this from the very beginning. Petra Frommer from Arizona State University, and these were quite large unit cells. These are membrane-bound proteins, and they were tiny. They were just a few hundred nanometers in size at that time. And so the fact that you see Bragg peaks out here to high resolution means that periodicity has to exist to that resolution. And if it's periodic, the unit cells are all the same, which means that they haven't exploded. Now, we could not only do that once, we could do it actually in that first beam time. We did it over 1 million times from many crystals. We sort of didn't know when to stop. And we didn't really know what to do with all this data because we were collecting it at 120 patterns per second. Of course, not all of them hitting a crystal, but we were shooting crystals through the beam. And Robert sort of showed how that worked. We had these liquid jets that were initially developed for kind of different experiments, but for molecular alignment experiments by John Spence, also at Arizona. And we had them just at the right time to sort of put into these experiments to do this. So there was really a lot of luck that came together to get that these experiments first worked. You also need the detector that can read every frame and read it out before the next frame. And of course, software to deal with all this data, which is something that, of course, we had to build at the time. So we've got many different diffraction patterns, each of a different crystal and a different random unknown orientation. And so we have to figure out what that orientation is, essentially index and figure out which Bragg peak is which. And then we can sum all this data in the frame of reference of the lattice rather than the frame of reference of the laboratory. And then essentially what we're doing in that case is sort of like powder diffraction, but where we've aligned every single grain into a three-dimensional common reference. But we're still averaging over all the sort of shapes and sizes of all those crystals. So each Bragg peak or each structure factor that we average is contributed to by a different set of crystals. And so we hope that each sort of subset of crystals is pretty much like every other subset of crystals. And so this method took off then and has been used for many different structures. Here's some of the early ones, including Cathepsin B, which was a target against African sleeping sickness, which Lars Redeker, my colleague, will be talking about later today. It was one of the early targets because we were looking for very small crystals to sort of try out the method. And he had these crystals of this particular enzyme that were forming inside living cells, which was quite cool. Now, many of these are sort of measured over and over. These codes here are depositions into the protein data bank. And these are where different structures have been measured in time, for example, or under different conditions, which now we can achieve because we have this very short exposure time, of course. This is just looking at these gas jets, well, liquid jets, I should say, gas-focused liquid jets that have evolved quite a bit over the years and are now a bit more, or certainly more reliable and usable. We have two fluids, usually one carrying the sample, the crystals, and then a sheath fluid that can be sort of have the right viscosity and surface tension and things like that to make this nice jet, and this works quite nicely. Of course, they have to be fast for the European XFEL because the pulses come so quickly. That's what Robert was showing. And likewise, you have to have a detector that can also read so fast. And they have this Egypt detector. Each pixel has its own set of storage cells. So each pixel can store 300 sort of values, 350, and then you read them out between these bursts. So you get 350 shots, pictures, frames per second. Per tenth of a second. So you get 3,500 per second. So it's quite a lot of data times a million pixels. So using that very high capacity, you can now kind of measure things fairly quickly. So this was in a few hours of beam time that Dominic Obertur in my group had to measure some insecticide proteins. So I thought this might be sort of interesting. These are natural insecticides. This is in collaboration with Colin Berry in Cardiff, looking at all these different forms that they're produced by sort of plants and insects, and the insects eat them, and when they get into and then they sort of naturally occur as crystals, and you can sort of use these as organic sort of pesticides, spray them on crops. When the insect eats them, they dissolve in the gut because of the change in pH, and they become toxic at that point. And so he was able to measure quite a number of these and new sort of structures. What's also interesting of this was that the phasing problem that I mentioned earlier that Max Peruch solved in one way can now be addressed using AlphaFold, this machine learning program from DeepMind that predicts structure just from the sequence. In this case, it doesn't predict it correctly. The prediction is in blue, and the prediction's in pink, and the final model's in blue. So it gets a little bit wrong, but close enough to use as a sort of starting point to do refinement, essentially refine all the positions of all the atoms that match the data. And so now with that, powerful tool, which was built upon having all those protein data bank entries to work from, now you have that, you can, we can sort of utilize that, and that will make these experiments go even faster and more reliable, and to be able to do sort of time resolved structures quite nicely as well. So talking about time resolved, we can look at photoactive proteins quite nicely. Just shine an optical laser onto the same region of the jet as the X-rays hit, and then for short time delays between an optical laser coming and the X-ray pulse coming, there's time that a reaction occurs. Usually in that time, the crystal itself hasn't moved very much along the stream. And so you can sort of do measurements just at that with that one particular delay, and then change the delay and do more measurements. And here's the photoactive yellow protein showing this first some sort of change in density. It's shown as this difference density, positive and negative. So negative where there was density and there is no longer, and positive where there is new density. And so you can sort of visualize motions in that way that are occurring around this chromophore. But then we're able to measure this at a much finer, shorter time delay. And here it gets tricky because you need quite an intense optical laser to do this initiation. And then there's worries about whether you're sort of bleaching your sample or having multi-photon effects and things like this. So it is quite still quite challenging. But nevertheless, you can sort of see this transition occur in a real sort of isomerization of this bond here within about 500 femtoseconds from trans to cis. Now, the fact that we also have this liquid delivery also means that you can look at sort of mixing reactions. And in this collaboration with Yunxing Wang from NIH, together with quite a large, all this work was done in parts of large collaborations. We're able to basically mix into this gene switch. It's a messenger RNA that has a particular structure and when it mixes with the sort of ligand that's involved in the protein it encodes, then there's a change in structure of the messenger RNA, this aptamer, which then prevents it from being read by the ribosome. So it silences this gene essentially through a structural change. And this change you saw was quite large, and yet it still occurs in these very small crystals. If you have a larger crystal, this change produces so much strain and change that the crystal just disintegrates. So it wasn't possible to sort of measure it previously, but now that we can look at these very tiny crystals, because we can expose them so much, we were able to basically overcome that issue. That the crystals go through this transformation, but seem to survive the process. They even change sort of space group, which is quite incredible.
5 minutes, Henry.
Okay. Yep. So just in these last few minutes, just talk about so I've talked about serial crystallography for FELs. Now, one of the realizations was that we weren't only sort of exposing these crystals much more than what we would expose a crystal using the old ways of having a single crystal in the beam and rotating around. We could not only increase the exposure, but we could sort of reduce the size and reduce the exposure. Now, this is a plot showing size of objects and kind of this exposure, which is proportional to dose and to strength of the diffraction pattern. And so these lines here are sort of for a given strengths of diffraction patterns So number of atoms or object size times the number of scattered photons per atom is number of scattered photons. So this is sort of what you would need to get a structure from a single object. Our experiments were able to, well, increase the exposure, but also decrease the size of the object and deal with much weaker patterns, and even go to lower exposures, to synchrotron sources, even sort of down to lab sources, but also to compact sources are very suitable for these types of experiments, because we can just measure many patterns and build up the data. And so one way that we've been doing this at the synchrotron, since you want to sort of expose it with the right time frame of a few milliseconds, you get enough you get enough exposure on your sample to essentially not expose it too much that you damage within that time, and enough that you can see diffraction from tiny crystals. And so you don't want the sample to move through the beam too fast, but just fast enough that it, say, transits a very intense focus beam at the synchrotron within a few milliseconds, as you see here. So what we're doing is essentially having this stream of liquid right onto a tape. You can sort of see this stuff moving along here. That's junk on this tape. It's just like a reel-to-reel tape of Kapton. You see it also here. This liquid has just been written onto the tape, which is moving across and dragging the crystals across. And then we measure with a detector at kilohertz or something like this frame rate, and get 1,000 diffractions patterns per second. And you can see that it's quite concentrated. So almost every shot does hit a crystal. And this way, we don't have to do things at cryogenic temperatures. So you can mix with different environments that would be usually quite bad for the crystals, but they only have to survive for a little time. So the approach of zero crystallography is quite flexible and can be used at pretty much all the sources. And so there are also many different ways to trigger reactions. So you can look at different types of reactions for time resolved measurements and really open up structural biology to the time domain. Also get it away from this looking at structural biology at cryogenic temperatures. It could also speed up screening types of experiments. There were many of those in the pandemic of trying to find drugs against various proteases and enzymes in the coronavirus, for example. So that could be really accelerated a lot. And there's many places to do this kind of thing. So I guess I'll end there. Just also acknowledge many people in a few groups, a combination of groups here of myself, also Sasha's group and Alka's group, working together on these types of things. But also to acknowledge many collaborators over the years that we've done a lot of these serial crystallography experiments at the FELs and synchrotrons. So thanks for that. I'll stop there.
Thank you, Henry. Very nice talk. So are there any questions? Is that an old hand I can see there from Lawrence, or a new one? Probably an old one then. Any other questions?
No. But I do have a new question if there's no.
Oh, you do have a new question? Okay, go on.
So there was a PhD thesis from Cornell at Chess, where they investigated the idea of implementing serial femtocrystallography in a normal MX beamline. Are you familiar with that work?
Not with the chat. I mean, there's quite a few places now. Serial crystallography is done at almost all the synchrotrons.
Yeah.
Yeah. So we let's see. So I mean, I know at APS they were doing things as well. There's certainly here at Petra and at ESRF. They've now even built dedicated beamlines for this. But I'm not entirely sure who was doing it at Chess.
The student's name was Jennifer Weyerman.
Right. This was okay.
I can't remember her name of her thesis advisor.
Yeah. So she was working with Veit Elser and also Saul Gruner.
Yes, exactly. Saul Gruner, yes.
Right. But they did some extremely nice measurements, and it's actually on this plot here.
Yeah.
Which was this plot here. That was with the lab source. I mean, they started even sort of smaller, but they were measuring big crystals with extremely low dose, and getting just a few hundred scattered photons per shot, using essentially an FEL detector at a lab source, and I managed you could understand all that data and treat it.
Okay.
Very good. Thank you. So let's thank our speaker in the interest of time. Again, thank you, Henry, for a very nice talk. And I think we'll go into a break now. So we have a break, and I think we'll reconvene at 3:50, if that's okay. So just over 5 minutes break, and we'll start back at 3:50. Is that okay for everyone? Let's do that then. Okay. Thank you again.
Okay. Thanks, Henry. Thanks, Henry and Robert, for doing this.
Thanks very much.
Okay.
Thank you all.
So the after break speakers are here. Gideon's here, Simon's here. And Lars sent me an email earlier. I guess he will be here. I don't see him at the moment. But who was the first speaker after the break, Marcus?
That should be Gideon now. So Gideon's swapping with Henry. So
okay.
If he's here, that would be great. We'll just check. Gideon, can you just let us know you're here if you are?
Can you hear me?
Oh, there you are. Wonderful. Okay, great. So good to see you. And
Sure.
yeah, we'll hear from you in a few minutes.
So thanks a lot.
Wonderful. Thanks. I'm going to move away from my computer just for a moment. perhaps we can start back up again. Is that okay, Lawrence?
Yes, sure.
Wonderful. Okay, so let's.
Gideon,
Yep. So our next speaker will be Professor Gideon Chinamantra Atira, from University of Witwatersrand. And he will talk about or provide a report on the LAMPS FAST team visit to the ESRF. So please go ahead, Gideon, and share your screen.
Thank you very much. Can you see my screen now?
I can, yes. Please go ahead and full screen.
Okay, thank you very much. I'm going to report on my LAMP FAST team visit to the ESRF in France, which happened between January and March of this year. So at Wits University, we have a laboratory-based computed tomography setup. And this is just a schematic to show how that actually works. So you're not getting a typical laboratory setup. We have an X-ray source that emits radiation, which passes through the source, which passes through the sample rather, placed on a rotating sample stage like this. And the CT is based on the principle that the density of an object, which is traversed by an X-ray beam, can actually be measured from calculations of the attenuation coefficient of the absorbing material. And we name this the attenuation coefficient, a measure of the quantity of the radiation, which is either absorbed or scattered by a given thickness of the absorber. And the beam that passes through these objects are projected onto a flat panel detector to actually form a projection, which is sometimes called a radiographic image. So this is a picture of our CT scanner at Wits University. So there are main three stages that we perform in a lab-based CT, which is data acquisition, image reconstruction, as well as image visualization and processing. So during the data acquisition is the step that I just described above, where we are measuring the transmitted beam intensities by a flat panel detector. And then at the stage of image reconstruction, this is a mathematical process which actually converts the scan data into 3D data. And then finally, the visualization and the processing is usually done by the end users of the data, which are by different visualization software such as Aviso, ImageJ, and other softwares that you can use for this. So you will note that at the stage of image reconstruction, there are actually several algorithms that can be used when this 3D volume is generated from these radiographic images. And one of them that has been used in these lab-based CT setups is the filtered back projection. However, it has limitations such as noise and prolonged reconstruction times. And it is a combination of actually these restrictions and the advancement of computers that iterative reconstruction have been seen as a possible method to replace this kind of image reconstruction. So here we used what is called the Astra toolbox, developed by a research group in the University of Antwerp in Belgium. It's actually still in its developmental mode, which means we had to do a little bit of development to this algorithm. So what it does is it enables you to use iterative reconstruction techniques in a lab-based CT. However, at this time, because of hardware limitations, we scanned a tooth, a fossilized tooth, and the image A shows the reconstruction of this fossilized tooth using the simultaneous iterative reconstruction algorithm using just about 300 projections, which was about a 10th of the full data set, as we were still testing the viability of such an algorithm. And then the reconstruction software that comes with the machine is the filtered back projection, and it's called CT Pro 3D. We also reconstructed it just to compare these two. And initially, the end goal is to develop data using about 3,000 projections of the Astra in the Astra toolbox using iterative reconstruction techniques to get a full data set. And we want to be able to improve this data. So the advantage of this toolbox that we used, we had to write a MATLAB code, and we had to develop some techniques such as center of rotation recorrection and beam hardening corrections as well, which we are still trying to work through. But actually, the advantage is that it has a modular setup where you've got the projection geometry and the projection data are separated from the volume geometry and the volume data, and they only meet at the projector, and they are subsequently performed in an algorithm which goes back and forth until you have enough iterations to produce a complete data set. So we are still working on this data to actually develop it. So at our lab, we are mainly focused on scanning fossils. However, at a university setup, we also serve the university, and we get researchers from different other fields coming, such as the industry, such as metallurgy, also material science coming to use the CT scanner. And time and again, we have to actually carry the specimens to the synchrotron to then perform better data. The synchrotron comes in the sense that in a lab-based CT, we are using absorption contrast, and at the synchrotron, we have the advantage of being able to retrieve or yield phase data from these materials. So during my time at the ESRF, the European Synchrotron Radiation Facility, I was at the BM05. This beamline serves as a test and development station, and is actually available for internal beam development and proprietary research. So we could use both the white beam and the monochromatic X-ray beam at this beamline, with the white beam ranging from about 25 to 120 kilo electron volts, and the monochromatic beam just about 8 kV to about 60 kilo electron volts for our experiments. So these are the physical components in the BM05, which we have. So here we have the optics hatch, which has got primary slits, the attenuators, and the monochromator. The job of the primary slits is to collimate the white beam. And you will note that these slits are actually cooled using a water circulation system. And we actually use them each time we're setting up for a tomography experiment, adjusting the horizontal and the vertical gaps that were within this. You also note that we've got attenuators, and these white beam attenuators are used for absorbing lower energy X-rays. And they're actually plates that are made of tungsten or copper or molybdenum, which had varying thicknesses. And we actually use them to adjust the energy of this white beam, depending on what energy we wanted to use. And then we had the monochromator, which is used to select the desired energy, which was actually used in attenuating the white beam. We actually did not use this for tomography experiments that we actually did carry out during our visit. We also have an experimental watch, EH1, which is not in this image because all experiments were carried out in the experimental watch 2. But we had a few components that we used in the EH1, which were the secondary slits that were used to further collimate the X-ray beam, and the first shutter, which actually protected the detector when no acquisition was being taken. And in the experimental hatch 2, we had the tomographic sample stage where our sample would be during the experiments, and with our 3 detectors, the D-zoom, the zoom, and the twin-mic detector. The twin-mic detector has discrete resolutions of about 1.4 microns, 0.7, and 0.35, depending on which objectives you use on the detector. The zoom and the D-zoom actually cover the continuous range from 1.2 microns to about 25 microns. So this is just to show you the image of the experimental hatch 2, where we had the sample stage and then the 3 detectors lined up from with different propagation distances from actually the sample. So we did carry out a number of experiments. The first, we had hominin teeth from Krondra in South Africa. We also had a fossil crocodile from Zimbabwe. It so happened we also had 2 researchers from Zimbabwe on the project, which was actually a very exciting thing to do. And we also had titanium alloys from the CSIR in South Africa, which were manufactured by the metal injection molding process. So this metal injection molding process, also known as the powder injection molding, is actually a cost-effective method for high production volume of high-density metal parts, which actually possess a complex shape in which we require a high dimensional accuracy. So this technique has been used or is used because it has high advantages such as high mechanical properties and higher production densities and a better surface finish than other powder metallurgy projects. So in this technique, which we had, you actually see that at the mixing stage, you've got a metal powder, which is mixed with the binder and combined to form a heterogeneous mixture. What then happens is that at the injection molding stage, injection molding machines are then used to inject this heterogeneous mixture to inject the green part. And once this happens, This picture, you see that it has a low viscosity and can be molded through the use of high pressure screw injection molding machines. After the molding, the binder is removed in a debinding process from the components by techniques such as thermal or a solvent debinding. And after removing the binder, they then perform a process which is called sintering at controlled temperatures and heat rate. So during this metal injection molding process, you will note that there are defects such as porosity that arise. And our aims in this project was to determine the spatial distribution of porosity in the injection molded volume at various stages during this processing, and also to calculate the porosity distribution of the metal particles, of the powder particles rather, and improve the metal injection molding processes. Other work that is still to be done is to determine the internal stresses, the composition, and the structure of this. We are going to actually employ diffraction methods such as 3D, XRD, and coherent diffraction images to actually carry out this research work in the future to add to the work that has been done so far. So this was our sample mounted, and here we were using the tie clip that we scanned here, which was made with the metal injection molding. What we did was we mounted it at a distance here, a propagation distance from this detector, which was here. So you will note that what we did was we performed a multi-resolution scan. Firstly, we scanned it at a resolution of 2.44 microns on the zoom detector, and we also scanned it 0.36 microns. The 20 times objectives was used on the twin detector, and also we scanned it at 1.44 microns. And then to reconstruct this data, we used a fast tomo to initially check the reconstruction, and then the slices were reconstructed to a full volume using PyHST software developed at the ESRF, which actually uses MATLAB and a filtered back projection algorithm. So this just shows the data of the multi-resolution scan. This was at 2 microns to the left, and to the right is at 0.362 microns. We are still to work on the data from the 1.44 microns. As you can note, initially the difference at this resolution, you actually can see that at 0.36 microns, we start to see some white material appearing in our data, which is suspected to be the phase shift of vanadium from the tie clips during the acquisition process. Further tests will be done to actually confirm these results. So what we did to analyze these results is we used image binarization just to separate the pore spaces which were within these tie clips and the metal powder to separate them. So we used like a threshold where we either had pore spaces and the metal powder, and we had a threshold value within the data set that we actually did have. And then to segment this data, to separate the pore spaces from the metal powder, or rather from the powder material, we used Dragonfly, which actually used a histogram basis analysis of the intensity levels for determining this threshold, which I actually mentioned above. And this Dragonfly software, which is available at the ESRF, has the auto algorithm, which was embedded in it, which actually retains a single intensity threshold, which helped us to separate these pixels into two classes, foreground and background, with our background being the pore spaces and our foreground being the powder injection molded particles. So what we did is we then took this volume, and to study the vertical heterogeneity of the porosity, we segmented this this tie clip into 10 equal volumes vertically of about 14.4 millimeters in height, which gave us a volume of about 16 cubic millimeters each of these volumes. And then we were studying the porosity in each of these volumes. And what we identified is that as we move from the top to the bottom of the tie clip, the porosity actually decreases. We had initially measured the average porosity of the whole volume, which we found to be around 7.27%. And then our range of the porosity was varying from about 7.53 to about 7.02% within the tie clip. And the assumption is that the material is more fluid during the metal injection molding process as the during the injection process is more fluid at the top and becomes more compact at the bottom during the injection molding process, and then hence the porosity actually decreases at the bottom as the material becomes more compact, unlike when it's actually more fluid, like at the bottom, so at the top rather. So we're actually able to determine the vertical distribution, the spatial distribution vertical of this porosity. And to add on to this, we also studied the horizontal, rather the radial measurement of the porosity, which we did what we did is we created cylinders. We segmented rather from the whole tie clip, small cylinders. Horizontally, we had about 8 cylinders, each of our radius 0.11 millimeters and about volume of about 0.5 cubic millimeters. And we also did the same vertically, radially, just to see how the porosity varies as we move on from the edges of the tie clip towards the center. So what we observed was that along the x-axis, the porosity varied from about 5, and then you'd find that the porosity, as we move a few millimeters into the tie clip, the porosity actually increases and then starts to actually slow down as well. The hypothesis at this stage is that the porosity of these tie clips actually lie a few millimeters into the tie clip, which is yet to be further confirmed by other measurements, but we are still basing on that hypothesis based on the results that we actually got. And we also did the porosity measurements along the y-axis, and we actually found similar results where the porosity actually lies a few millimeters into the tie clip from the edges, and then it starts to go down. Like I mentioned with the x-axis measurements, that are further tests such as pore size and pore distribution are to be made to actually determine how and why the porosity actually lies a few millimeters, if it is true. Actually, that's the conclusion. And then we also worked on hominin teeth from Chrome Dry in South Africa, like I mentioned before. This project is done in collaboration with Professor José Braga from the University of Toulouse in France and Professor Francis Thackeray, together with other collaborators. So you will note that incremental growth lines that manifest on the crown surfaces after coursing through the enamel are used to determine the growth periods at the edge of death for these teeth. And they are formed by enamel secreting cells during tooth growth. And in the past, the process required physical sectioning of the tooth before actually analyzing these slices through the main cusp axis of the tooth with the use of a microscope. But with the use of propagation phase contrast computed tomography, we were then able to do a non-destructive imaging of the internal aspects of these dentine tissues. And what this part of the project seeks to do is seeks to quantify the dental long period growth lines and matching stress percent, which actually be then used to determine the crown formation at the time of death in hominin fossils from South Africa. So if we then combine the 2D virtual histology and the 3D high quality rendering of these dental surfaces and interfaces, we are then able to actually quantify these long period lines and developmental defects which are actually observed. So we did this project, and I think Dr. Kathleen Dolman from the ESRF will shed more light on this in your presentation tomorrow. Also, we worked with her on the fossil crocodile from Zimbabwe, and this project was done in collaboration with Professor Shonay from the Evolutionary Studies Institute, and Dr. Kathleen Dolman, who I mentioned, will also give a presentation tomorrow. I'm sure she should touch some light on these experiments with fossils and paleontology on this data. But with this fossil crocodile that we studied, what you note is that this specimen represents only one of a very few terrestrial visible fossils that from a time period that we actually know very little of. And unfortunately, it was very, very fragile. So we had to actually wrap it up and carry it up. And we actually, this is even shows me actually setting up for a scan that we did. And we hope that this information what we get from this data, once the analysis is done, will be used to corroborate the Zimbabwean biostratigraphy with the main Karoo basins in South Africa is doing this research. So this was mainly the experiments that we did during the visit, and it was really a great time. Also being able to bridge that gap between a lab-based CT and knowing what we can actually do at a synchrotron. And also, thanks to them and the first team, I also had a great time in France. It was my first time to see snow, so I got to do some skiing. And I also was able to visit the Eiffel Tower and also just see around the Parc des Princes, the home of the Paris Saint-Germain football club. But yeah, that's all from me. I'd like to thank the following people: Ronald Machaka, Dr. Kevin Doman, Philip Cook from the SRF, Professor Shone and Professor Jose Braga, and my supervisor, Dr. Kudakwashi Jakarta, who was also here the SRF with me to assist to actually carry out this project and to have more information about how this beamline works and how best we can actually work on this, how we can work on phase contrast computed tomography imaging methods. Thank you very much. That's all from me. I'll leave this time for questions.
Thank you very much, Gideon. Very nice talk. Any questions? Please raise your hand or put in the chat. While I'm waiting, I have a question about the data analysis. So obviously the tomographic reconstructions will take some time. How much time do you normally spend on this, and what resources do you normally need to conduct the reconstructions? Is it just a PC or something similar?
So you mean the data reconstruction in our lab or the Israel? In our lab?
In your lab, yeah.
Okay, so yeah, for the floated back projection, which is the proprietary software that came with the machine, we just require normal PCs. But as we are trying to improve our lab and introduce iterative reconstruction algorithms, we actually need to acquire graphical processing units, which was one of the limitations why at this stage we're only able to use about 300 projections. But we are in the process of actually trying to acquire a machine with a bigger GPU size so that we can be able to actually do that. So in terms of the reconstruction time, using the filtered back projection, the normal reconstruction time is about 30 minutes to 45 minutes, depending on the data sizes. Also the, depending on the data size, the number of projections that you get during acquisition. And then with the iterative reconstruction technique, we actually expect a very big reduction in the reconstruction time, just basing on the experiment that we did with 300 projections. We actually almost half down the time of the reconstruction. So if we were to manage a very busy lab, this will actually assist us also for high volume data production.
Right, so it would increase your throughput in the lab, presumably.
Yes, definitely.
Excellent. And then you can be able to go up to maybe double the number of slices or more. What would you increase it by?
So we are actually hoping to actually use the full data set for the reconstruction, because we want to be able to match what is being done by the floated back projection at the full data set and actually improve it from there. So we are still in the initial phase. We're testing the viability, which is why we had just to use 300. But the goal is to actually use a full data set and then be able to deal with, number one, the reconstruction time that I mentioned, as well as the noise that is introduced by the floated back projection.
Very good. Any other questions from anyone?
Do we have time for a 2-minute follow-up video for what he discussed?
I think we do, yes.
Okay, could I share my screen?
Go for it, Sakazi. That sounds like Sakazi, yes.
Yes, okay.
So should I stop sharing?
Yes.
Okay, I just stopped sharing my screen.
Yeah, go for it.
Okay, Open system. I don't understand what's happening. Boy. Let me put it in the chat and then you can show it, because I'm having, I don't know why mine isn't working. Send it to the wrong person. Where is it, everybody? Okay, there it goes.
Gideon,
So
do you want to try and play that?
Sorry?
you want to try and play that? Do you want to play
Yeah.
that now, Sakazi?
Yeah, why don't you do it? I put it in the chat, and for some reason it didn't work for me.
Oh, okay.
Are you going to play it?
If you get it first, go for it, Gideon.
Okay, let me just see if I can play it actually.
Yeah, I thought this would be a nice follow-up for what he discussed. And it's only like a minute, a little over a minute. Having problems?
No, just got it. I just need to share my screen now. There you go.
Is there any
Yes.
sound on this? I can't hear any sound.
Yeah, it should be a sound.
So you don't have sound?
We can't hear any sound, no.
No,
You have to unshare and then reshare, and there's a button you can click to share your computer audio.
Okay.
I see.
Marcus, can you try to play it, please?
Yeah, I'll try. Just wondering if I'll be able to do it successfully. Just a moment.
My name is Gideon Chinomatia. I'm a postgraduate student at Wits University in South Africa.
I can hear something.
Yes, I could hear you as well.
You want me to try it?
Yeah, Lawrence, that might be helpful. I'm just mindful of this whole thing crashing.
So let me.
Has set up this kind of grants.
Are you seeing my
to
screen?
assist students
Yes.
and researchers in particular ways.
Let me start it from the beginning.
Yeah.
Yeah, we can see.
My name is Gideon Chinomatia. I'm a postgrad student at Wits University in South Africa. I'm here because of the LAMP grant. The LAMP has set up this kind of grants to assist students and researchers in particular where synchrotrons are not available to actually come and get a training on the use and the application of synchrotrons. For example, in this case, in Africa, we do hope to have a synchrotron in the future. So this kind of experience will give me a baseline to be part and parcel of the people who are actually going to be developing the use of synchrotrons and evolve research in Africa. Getting here, you realize it's the next stage of tomography. You actually start to appreciate the difference in what we get on a lab-based study and on synchrotron-based study. I was actually able to do some experiments and be taught on how to operate the machine. This is one of the samples that I actually scanned. I brought it all the way from South Africa. Unfortunately, it's very, very fragile and I can't open it. This is a crocodile, actually from the early Jurassic. It's the only known one from Zimbabwe. It was quite an interesting fossil to study. For anyone in tomography, this is the dream destination. You want to be here. You want to see how things work. You want to see how the data comes, because for most of us, we are the end users of the data. It's very easy to refer someone to the synchrotron when you have a feel of the synchrotron, when you understand what's exactly going on there.
That's very nice. Thank you for sharing that, Sakazi.
Yeah,
it's very nice to see that.
Proud of what they've done. Thank you.
Thank you.
It's very impressive. We'd like to see more of this type of interaction between the synchrotrons and research groups across Africa, of course. So it's very nice.
I put a question in the chat, which we can come back to during the Biophysics Week conference in March. But that one tooth that you showed that had the big cracks in it, what kind of animal was that from?
Hi.
Yes.
Yes. Hi. So the tooth is from a fossil. I don't remember exactly which fossil, but the reason why it doesn't look too good is especially when you look at the first two images, it's we're using 300 projections. Remember, not the full set of projections, the 3000.
Okay.
Yeah.
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 That cavity looked very uncomfortable.
Yeah. So in the cavity, yes. So in the cavity, you see, one of the other things that we're trying to look at is occasionally you find that in the fossils, when they're preserved, over time, you get some metallic elements sort of diffusing into it, and they kind of lodge themselves in some place. So this particular tooth has got some very high density material inside the cavity, which got in. And yeah, so that's where that glow in the center is coming from.
That's interesting. Thank you.
Yeah.
Okay, so let's thank our speaker again, as we can, with the claps, with virtual claps, and we'll move on to our next speaker. Okay, so thank you, Gideon. Again, it's a wonderful talk. Our next speaker is Professor Simon Boone, and he's from Columbia University, and he will talk about ultra-fast atomic per distribution function analysis, a new chapter in the goal of atomic movies. Hopefully you're there. Yes, I can see you, Simon. Please go ahead.
Yeah, thanks very much, Marcus. Can you give a thumbs up if you can hear me? Can you hear me?
Yes, we hear you.
Yeah, good.
Your microphone is working. Or unmuted.
Okay. Well, thanks very much.
I still can't hear you, unfortunately.
You can't hear me. One sec.
I can hear you, Simon.
That's weird.
Still the same.
I'm not sharing. Am I still? No.
Can you hear him now?
How about now?
Oh, yes, I can hear you. It's just going in and out. Maybe your mic just needs to be lifted slightly higher.
Okay.
Yeah, there we go.
I did actually change my settings. I think we're good.
Wonderful. Go for it.
Wonderful. Right. Thanks so much, Marcus. Yeah, thanks very much for giving me the opportunity to talk here and the invitation from Lawrence. I really appreciate it. Actually, Lawrence asked me to talk about ultra-fast studies that we're doing at LCLS, which is the X-ray free electron laser in California at SLAC at Stanford University. So that's what I'm going to talk about. And those of you who know me know that I've actually spent a lot of time. Let me see, I have to move this thing so I can put in presentation mode. Okay. Okay, those of you who know me know that I've spent quite a lot of time developing methods to study local structure using this atomic pair distribution function analysis. And our goal in these studies is actually to take PDF studies to the picosecond and sub-picosecond time-resolved domain. There are challenges there, but we've made a start, and that's what I'm going to talk about. And really, the scientific goal is in the direction of making atomic or molecular movies. So if you know the structure of a material, and Henry mentioned this, if we could in principle image atoms, then we could make movies of how they move under excitation. We don't have the ability to image them directly, but using diffraction methods, we can find where they are. PDF is a diffraction method, as I'll tell you, for studying local structure. So the ultimate goal is actually to have to watch atoms moving after they've been excited. Okay. Yeah, so this is just to say it's a great pleasure to be part of this wonderful effort to bring synchrotron light to Africa. I really strongly support this and advocate this, and I'm really glad that you're all here on this call. I also want to take the opportunity to mention that another activity I have with Africa is this JUAMI. JUAMI is the joint undertaking for an African Materials Institute. And every few years, we run a 2-week course for early career scientists in Africa. We focused on East Africa. And I just wanted to let you know that the next school will take place in June of next year, 2023. It'll be in Nairobi in Kenya. And applications are open, actually, for PhD students and young faculty in African universities with a preference actually for East Africa, but anyone can apply. And so please go to this website, jeremyjuami.org, if you're interested in applying for that, if you're on the African side. And these schools are great. We bring in international speakers. We have hands-on demonstrations. You work in teams with US PhD students to do problem solving and things like this. There's a video at this website that gives you the idea. So yeah, let me encourage you to apply for that if you're interested. All right, so the first statement that's fairly obvious to everyone is that the atomic scale structure is a sine qua non for understanding materials, and that's because the properties of materials depend sensitively on the structure or the arrangement of the atoms. This is just a graphic that I stole off the internet, which kind of illustrates this, where people have looked at different structural and morphological aspects of carbon nanotubes, and you can change their properties from being metals to being insulators, to have different thermal conductivities, and so on and so forth, just by small different rearrangements of atoms. And so knowing the structure of materials is a prerequisite for understanding their properties. And if we want to study what the structure does under excitation, looking at non-equilibrium structures, and if we want to do it in the ultrafast domain, we have to be able to study structure in this ultrafast domain. So there's a fairly long history of people doing ultrafast spectroscopy. This is a laser pump, laser probe spectroscopy. So you hit a sample with a laser, and then at some small time delay, you hit it with another laser. And the laser, the pump excites, but the probe actually studies the electronic energy levels of the resulting intermediate states. It's really doing spectroscopy. So this is a setup. You have a sample, you hit it with a strong pulse, you have a delay, and then a second probe pulse comes in. And this allows you to find the kind of quantum states of intermediates as the material undergoes some kind of change due to the excitation. But what you're seeing here is the energy levels. You're not actually seeing what the intermediates look like. And so really, a goal that we have is to see exactly how the atoms move as it goes through these intermediates. If you have ways of modeling, you can extract that out in a backhand way. You can use quantum mechanical calculations to get atomic arrangements at the intermediates, but we would really like to see them directly. So to see atoms, we want to use diffraction, and this can be diffraction of X-rays or electrons, also neutrons, although neutrons are not really relevant in this ultrafast domain. And this is a fairly old picture from this nice review article about, it was from 2010, but this is how you do this laser pulse X-ray probe kind of measurements. So you have to have this pump laser, which is going to produce an intense laser pulse, and it has to come down with a kind of femtosecond width and then hit our sample. And then after some time delay, we want an X-ray pulse to come down the tube and hit the sample. And then we'll analyze the X-ray diffraction pattern from that. So in the early days, people were doing that at X-ray synchrotron sources. People are still doing it at X-ray synchrotron sources. You don't have such good time resolution as you do at the X-FELs. But the synchrotron will typically put out a train of pulses that look like this. You put a chopper in the beam that will select individual pulses with some kind of time separation. And then you have a timing thing where your laser pulse is actually timed to the RF cavity of the synchrotron so that you can synchronize with some fixed time delay the pump and the probe. So this is the general setup, and it's been under development. This was a 2010 review, but it's been going since the late 1990s. So there's 20 years of development that's gone into that. Of course, what we have now is X-ray free electron lasers, and we heard about that earlier in the session. And these are exquisitely well adapted for this kind of measurement. And many ultrafast time resolved studies in the area of materials science have been rather semi-quantitative. And this is an example from our own work, actually. This is a collaboration with with a group in Korea and also Brookhaven. We simply melted gold. So what we did is we put a sub-monolayer arrangement of gold actually a gold film basically on top of a silicon nitride substrate, and we hit it with a laser. The laser is intense enough to melt the gold, and then with this time delay, the X-ray pulse comes through. So melting seems like it should be fairly boring, that we know that if you heat things up, they melt. But it's quite amazing that when you go into this ultra-fast time-resolved domain, you can really get new insights into fundamentals of material science like melting. So essentially what we saw, which was a complete surprise, is this is a Bragg peak, the 111 Bragg peak of the gold that's sitting at room temperature. And in the negative time domain, so the X-ray pulse is arriving at the sample before the laser pulse, that's all we see. So these blue curves here are all sitting at the position of the room temperature gold. What we expected to happen was that the intensity of this Bragg peak would come down, and then a very broad liquid feature would grow up, and we wanted to study the dynamics of that. What we actually saw was the second Bragg peak growing up. So these two Bragg peaks were coexisting, and the second Bragg peak grew up at a lower Q value than the room temperature one. So if you shift a Bragg peak to lower Q, it's like having a lattice expansion in the material. So it looked as if we had material that was coexisting, that was cool and then hot, but not melted. There is actually a liquid signal down here at the bottom that's not able, you can't see on this scale. But this was a complete surprise. And then we had to actually kind of come up with an explanation for it. But the explanation, and what we found was that the temperature of this second peak didn't change, like the position didn't change, but the amplitude kind of shifted. And the basic understanding we had is that the laser is dumping heat into the system. It's basically exciting the electrons into an electron gas. They propagate through the material very quickly, but then they get scattered by the grain boundaries in the film. And so most of the heat is deposited at this grain boundary region. And then what happens is you get a region that's very hot, but not melted. And then the melting happens on one side of that, and then that hot region just propagates in, and as the thing melts. So the dynamics of melting, it turns out, is surprising, and you can learn things that you didn't expect looking in the picosecond time domain, basically. Now, what we're doing here is we're doing laser pulse X-ray diffraction, and this is very characteristic of many studies in this domain in material science, where you'll actually extract a Bragg peak and you'll just look at the dynamics of the Bragg peak like this. We're not really getting molecular movies. In order to solve the structure from crystallographically, you would need to measure the structure factor of many hundreds or even thousands of Bragg peaks. And that's a much more difficult proposition than simply measuring the time dependence of a single Bragg peak. Another approach is to use atomic pair distribution function. I'm going to introduce it later, but this is another kind of fairly early study, 2008, which was doing time-resolved structural dynamics, where you take a molecule and you photoexcite the molecule. So this is not my work, it's this group over here, which is Michael Wolf, I know, but the collaborators are Korean. You start with this triiodide material, and what you see is that the first step is that you get into an excited state. The second step is one of the iodines detaches. And then if you do that to two of these, you have two individual iodines, and then they actually form an iodine bond. And these are actually the diffraction patterns as a function of Q, and this is the PDF as a function of R. And they were able to actually model the molecular intermediates and see the populations of them and the time dependence of them, and get kinetic information. So if you want to study kind of chemistry, and these are kind of molecular chemistries that are taking place in a solution, you can do it. But again, the resolution of these PDFs is very, very low. And the only way to extract the structural information was to actually have quantum mechanical calculations of what the intermediates looked like. So again, we're not quite at the point where we can do molecular movies. So what do we have to do to get closer to the molecular movies? So we want to actually develop the ability to do PDF in a much more quantitative way than has been done up to now. So if you're not familiar with the PDF, it looks like this. If you have a material that's isotropically scattering, so it could be a liquid or a glass or a powder of crystallites, and you send a beam in and you measure the intensity as a function of angle, you get a powder pattern, powder diffraction pattern. And if you make the right corrections to the powder pattern and the proper normalizations, then you can actually apply a Fourier transform and you get the PDF, which is shown here. So the PDF on this axis is a probability, and on this axis is a distance r. And what this encodes is the probability of finding two atoms in your sample separated by this distance. So there's a strong peak here in this material, it's 2.75 angstroms. There's a high probability of finding atoms separated by 2.75 angstroms. And all of these sharp peaks, which are on nanometer length scale, are telling us all of the intratomic correlations in our material on this nanometer length scale. We typically collect data to do PDF analysis from synchrotron sources. The experiments are very easy and we apply them to many, many different systems, from physics to chemistry to pharmaceuticals. The sample goes here, the beam comes down the pipe, and we have a very large area 2D detector. We get a scattering pattern which looks like circular rings on a detector. These are the bischerer cones that are intersecting the detector. These are the correction steps, and then at the end we get this 1D PDF. And then the next step is to extract structural information from the 1D PDF by modeling. So we can calculate the PDF from a model by placing an atom at the origin, moving out in R some distance, and whenever we find a neighboring atom around the origin atom, we place a delta function there, and we broaden that delta function with a Gaussian to account for thermal motion. And then we go out a bit further, we find another ring of atoms, and further, and we can build up structural models if we can calculate the PDF from structural models. And then we can do regression, so we can do fitting. So we can vary parameters in the structural model until we get a good agreement with the data. And this figure here in the blue curve is a measured PDF of nickel, and the red curve is a calculated PDF of nickel. And this is the level of agreement that we can obtain. So we're really obtaining the three-dimensional coordinates of the atoms in our sample in this case. Just to briefly mention, we're able to do this with X-rays and neutrons, but also with electrons. So this is a PDF obtained from an electron microscope. And the reason why I'm mentioning this is that in principle, we can do ultra-fast electron diffraction to head in the direction of molecular movies with a laser pulse X-ray probe. These were gold nanoparticles, and you have a very small sample. So the actual sample are these tiny black dots. These are the gold nanoparticles that are sitting on top of a holy carbon film, but they give a good diffraction pattern that can be corrected, and then we can get, again, quantitative fits to the data. It should be mentioned that the quality of the data from the electrons is not quite as quantitative because of the dynamical scattering effects of the electrons. But nonetheless, we can get very high quality PDFs from X-rays, and we can get medium quality PDFs from electrons. Okay, so what's the secret source of PDF, and why can't we do it right now at an XFEL? And really the reason is coming from this momentum transfer Q. So Q is actually the difference between the wave vector of the incoming wave and the outgoing wave. So Q is actually K naught minus K. It could be defined either way, but usually K naught minus K. And that gives us Q. And in order to get a very high resolution, real space resolution PDF, we need to measure Q up to very high momentum transfer, a very high value of Q. And in order to do that, we need very short wavelength X-rays. So this is an example of data that we get from cadmium selenide nanoparticles. And this Q value here is taking us up to 28. And it looks like there's no signal out here. It looks like the curve is very, very flat. But we have to divide by the form factor squared of the X-rays. And when we do that, we get a signal that looks like this. So taking you back, if you just look at the raw data, it looks like there's no signal out here. But when you look at the properly corrected data, there is a signal out here. And it's actually this high Q signal which gives us the high real space resolution when the thing goes through the Fourier transform. So to get highly quantitative models, we want to measure to high Q. In order to measure to high Q, you need to use very short wavelength X-rays. This is an example of the different quality of data. This is a copper K alpha lab source measuring a nanocrystalline material, and it basically only has 4 bumps, and it goes up to a 2 theta angle of 40 degrees with copper K alpha. And if we do the proper PDF experiment up to high Q, this is the same signal in the low Q region, but we get all of this beautiful data out to high Q, and that's what allows us to do the quantitative PDF analysis. So we love X-FELs because they give us these ultra short pulses and these very high intensities. But even the hard X-ray X-FELs up to now have not given us X-rays that are short enough wavelengths to do PDFs. This is just an illustration of here of what I showed you the nickel PDF earlier. This is again the nickel PDF with a Qmax of 30. And if we look at the same function with a Qmax of 8.5, which is what can typically be obtained in these kind of early X-FEL and time-resolved synchrotron experiments, you can see that the peaks are very broad and overlapped, and there's a huge loss in the signal fidelity. So this is one of the main challenges, which is why we don't have ultra-fast time-resolved PDF now. But things are changing. So LCLS2 is coming online now, and it has actually higher harmonics, which are sufficiently high in energy and short in wavelength with good intensity that we can start to think about doing PDF measurements. And on the horizon is LCLS high energy, HE, which is the construction is planned, and there are also plans for high energy X-FELs in Europe. So I think that the future looks bright for doing PDF. With that in mind, then we decided to attempt a fully quantitative, ultra-fast, high real space resolution atomic PDF study. And the rest of the talk, I'm just going to tell you about this study. So we're physicists, and actually we wanted to study this system, copper iridium sulfide, which I'll tell you about shortly under photoexcitation. The work's not published. We're still analyzing the data, so this is just a progress report. So we've been studying copper iridium sulfide for a number of years. It's actually one of the big projects of Elm Bojin, who's my collaborator at Brookhaven. The samples were made by John Mitchell at Argonne, and it's got a rather complicated structure. It's a spinel. But what's interesting about it is it has metal iridium ions that sit on the pyrochlore sublattice. And the pyrochlore sublattice is a set of corner-shared tetrahedra that looks like this. And at high temperature, this is an undistorted pyrochlore lattice. All of the iridium-iridium bonds have the same length, and it forms a cubic structure. At low temperature, though, the iridiums dimerize. So some of the iridiums choose to form little kind of short bonds with each other, and others keep long bonds. So what this looks like in the PDF is we have our average iridium-iridium distance at 3.5 angstroms. And then that's in the cubic phase, that's the only peak that's present. But in the low temperature dimerized state, this thing separates into having a short iridium-iridium distance at it's actually half an angstrom shorter, like 3 angstroms. And there's also a long iridium-iridium bond that forms, and some of them are still at the average distance. So we've studied this for a number of years. It's a very interesting quantum mechanical effect. It comes from charge ordering. It's related to a metal insulator transition. You don't need to care too much about that if you don't care about it. But the point is that at low temperature, quantum mechanically, these iridiums want to form these dimers. They're a little bit like covalent bonds on the lattice. And we can see this dimer form in the PDF. So we can really see this effect in the PDF. If you just increase the temperature, the dimer goes away. So this is the dimer peak at this position here. And then at this temperature, which is around 190 Kelvin, that dimer simply goes away. And what we want to do in the ultra-fast domain is we want to actually cool our sample to low temperature, so it's in the dimer state. Then we want to hit it with a femtosecond laser to excite the system. Now, what we're hoping is that the excitation will actually destroy the dimer. It will cause the dimer to go away. We don't want to melt the sample this time. We just want to look at the dimer state dynamics. We would love to answer the question, what's the resulting state when the dimer goes away? So what's the non-equilibrium excited state of that system? We want to know how quickly the dimer state is destroyed. It's an electronic state, but it also involves lattice relaxation. The atoms have to move to the shorter distance. So how quickly does that go away? And if that goes away locally, if I destroy a dimer locally in my lattice, how does the rest of the lattice respond to that destruction of the dimer state? In other words, how does the effect propagate through the lattice? So this is a little bit like what's happening in a tsunami. In a tsunami, you have an earthquake at the bottom of the ocean. That's a fairly localized event. It's happening at one point in space, but it produces a disturbance which then propagates out through the propagation of waves, in this case, water waves on the surface of the ocean. And that actually takes energy and deposits it in locations that are far away from where the initial thing occurred, the initial earthquake occurred. And that takes some time for that wave to propagate out. And it's exactly the same thing we could imagine happening in our copper iridium lattice. We've got a dimer, we destroy the dimer locally, and then how does the strain wave propagate through the lattice? And then what's the lattice response to that? So these are some general questions that we had that we hoped could be answered by this ultra-fast experiment. So the experiment was done at the MFX beamline at LCLS, which is at the Stanford Linear Accelerator Center. Because we don't have LCLS HE, we were limited actually to a modest Qmax. But again, we're just trying to solve the problems of how to get quantitative PDFs, and we're doing this in preparation for higher energies. We used an 800 nanometer titanium sapphire laser. There's a delay line built in that allows us to synchronize with different delays to the X-ray pulse. So this is the basic setup. Our sample is here, it's being cooled by a cryostream, and then we get our diffraction pattern. Actually, the black powder was put on a Kapton film, and so this was the final geometry of our setup. The data reduction has gone actually more smoothly than I expected, but we've got actually good methods now for analyzing the raw data, getting it into 1D diffraction patterns, turning these into PDFs. And so this is actually a very beautiful time-resolved set of PDFs from the experiment, and they're clearly highly reproducible. They have reasonably good spatial resolution, and we can see some small effects happening, but the reproducibility is clear. So we were very happy with this initial view of the system. Of course, we want to see what the changes are and which changes are important, and so we can focus into that. As a sanity check, of course, what we're interested in is the destruction of the dimer, and at room temperature, the dimer doesn't exist. So if we do the whole experiment at room temperature, we should basically see nothing, except for possibly some kind of thermal expansion effects and all around boringness. And basically that's what we found. It's very noisy, and the reason it looks like this is just because it's plotted on a very expanded scale. So there's a small amount of thermal expansion, but there's nothing very interesting. But if we hold our sample at 150K, we're in the dimer state, and we do the photoexcitation. These are difference PDFs. So we're taking a difference between the PDF measured at time t and the PDF measured at time at a negative time. And what we see here is we actually see a dimer signal appear almost immediately, in fact, on 1 picosecond. So remember what the dimer signal would look like. It would be we'd have the presence of the dimer, which is a short and a long bond here, and the short and the long bond would disappear, and they would be replaced by an average bond. And that's exactly what this W shape in the difference curve looks like. There's intensity that was here and was here before the experiment, ends up here after the photoexcitation. So this is exactly our, okay, thank you. This is exactly our dimer signal. And then you can see as a function of time, as a function of distance in R, it propagates out, and as a function of time, it actually grows and increases. And so if you look at the pale blue curves down here, it's a little hard to see, but that's what the dimer excitation looks like at high R on the almost instantaneously. And then as time progresses, the strain, this is the tsunami effect where these amplitude peaks increase over time. I just want to mention that we were able to quantitatively model this PDF. So this is actually a fairly complicated structure of the copper iridium sulfide, and the fit quality is somewhat comparable to what we can get from actually regular PDFs. So we're getting beautiful quantitative PDFs from this LCLS setup. What we found was that the quality of the fit, so this difference curve can be encoded as an RW, a weighted agreement factor, and we have very good low weighted agreement factors at low times. But then as the time progresses, the agreement gets worse and worse. It's actually worse in the high R region at high times. And that's because there's an excited state, which is actually not well captured by our model. So that's exciting. It means that there's a signal in our model that will tell us about this intermediate state. And at this moment of time, we haven't solved that structure. So I can't tell you what that intermediate state is. But again, just looking to the future, PDF modeling can extract quantitative local structures in principle. And an interesting thing for me was that actually the dark blue curves are negative time, and they just, that's the kind of level of reproducibility we have when we measure the same structure with multiple different pulses. And the pale blue curve is the first the first positive time differential, and already we see this, it's a little hard to see, but already we see this dimer curve within 1 picosecond. But interestingly, we also see signal out to high R. So what it tells you is that the quantum mechanical wave function for this dimer state is actually not localized to a single point on the lattice. It actually extends, but it only extends up to 3 or 4 nanometers. And so this is something that we're going to investigate further as time goes on. And then we can extract actually multiple different components in the time signal. So there's a very, very sharp time component, which is the destruction of the dimer, which happens on a time scale faster than our time resolution, less than 1 picosecond. And then there's this slow effect, which happens because this tsunami effect, where the dimers, the strain from the dimer destruction propagates out through the lattice, and that happens on time scale of up to 100 picoseconds, and it's still evolving. Okay, so I tried to give a general introduction of ultra-fast time-resolved structural analysis with a view of how PDF analysis can play a role in this game, with the goal of making molecular movies. We thought we were going to encounter challenges, and we did encounter challenges, but largely speaking, it's been quite a successful foray into this. And with the advent of harder X-ray XFELs, this is really going to turn into a promising avenue of study. Here are some of the challenges, but basically the future looks bright and fast, I would say. So with that, let me thank you, the audience, for listening. This is a picture of my group some time ago. These XFEL experiments involve large numbers of people who make samples and do the experimental setup and do the timing and run the laser and so on and so forth. It's a really wonderful way to do collaborative science. And coming back to the African context, I think maybe a synchrotron before an XFEL, but whether it's a synchrotron or an XFEL, it really fosters community-based science, which is one of the truly truly nice things about it. And with that, thank you very much. This is what my group looks like in Zoom times.
Very nice. Thank you very much, Simon, for the very nice talk and for the images of your group. Great exercises. So that's great. Not sure I can move like that. Anyway, so any questions from anyone? I have a question actually. So regarding the dimerization of the iridium pyrochloro sublattice. So what actually drives this? So is the sublattice non-centrosymmetric?
Well, so the high temperature, it's symmetric. It's a cubic. What's driving it is the question that we're trying to answer in some sense. But there's a lot of things that happen together. The first thing is that if you look at the pyrochlor sublattice, it's this really kind of three-dimensional thing. But when the material forms dimers, it forms them along these one-dimensional chains. And so it's electronically driven, basically. It's a little bit like a charge density wave. If the material, these are nominally iridium 3+, 3.5+, and what the material does at low temperature is it forms a charge density wave, which is iridium 4+, 4+, 3+, 3+, 4+, 4+, 3+, 3+, and then the 4+ ions dimerize and the 3+ ions separate. And so it's a kind of a similar driving force probably as charge density wave formation, like Fermi surface nesting or something like that. So bottom line is there's an energetic driving force for it to do that. We don't know exactly what it is, and there are multiple things going on. But yeah, that's kind of the origin of it. Okay, so is there a ferroelectric phase as well in this? No, this thing doesn't break centrosymmetry. Right. Yeah, that's an interesting question, though, whether we could actually induce that, because the thing about ferroelectricity is that you want to break centrosymmetry, but you also want to have these kind of preformed dimerized or preformed polar domains that you can then form into large domains and pin to get the ferroelectricity. So these things are all certainly related. Very nice. You also mentioned wanting to go to higher Q in your measurements. How long is that piece of string? So you mentioned 10 for example, in inverse angstroms. What would be ideal for you? So the most high quality PDFs for most cases for hard materials are around 30 inverse angstroms. Okay. And we can get that at synchrotrons routinely. Many of our experiments are done less than that, like 23, 25. And for organics, we can go down as low as 12 or 15. So we're not far away from, it really depends how soft the material is. So for very rigid materials, you have to go to higher Q. For softer materials, lower Q. But I think the sweet spot is kind of 20 to 25 inverse angstroms, and the 30 is even better. Very nice. Any other questions from anyone? Please raise a hand. Put in the chat. No? Okay, well, I think we'll then just thank our speaker. Thank you very much, Simon, again for the nice talk. And we'll move on to our next and final speaker. So that will be Lars, Professor Lars Radeke. Thank you. And he's from the University of Lubbock. And he'll talk about in vivo crystallization of protein targets for African sleeping sickness therapeutics. So over to you. Lars.
Hello, I guess you can hear me?
Yes, I can.
Oh, that's great. So start sharing my screen. Give me a second. So I hope that you can see my screen now.
Yeah, we can see it.
Oh, I think I have to. Do you have the presentation mode or the other one?
The other one, yeah, if you can switch.
Yeah, I have to switch. Give me a second.
It's at the top, in the middle, very top. It's Ansingen at the very top in German. In the middle.
Very top.
Yeah.
Ah, they're
On the
there.
left-hand
Yeah, I
side.
know.
Yeah.
I know.
Yeah.
Yeah, perfect. Now you should see it. Oh, perfect.
Yeah.
Now it runs. Yeah.
Yeah, there you go.
So we can go. Yeah. Thank you for inviting me to present our results, which are of course related to XFELs, but also to synchrotron sources. So we use both of them. We have to switch the topic now, I guess. So we will focus now on proteins. But as I understood, you would have you had already quite good introduction today by Robert Weigenhansel about the European XFEL, and also by Henry Chapman, of course, introducing you into serial femtosecond crystallography, which will also be an issue in my talk. So I hope that I can build my talk on these great colleagues. Yeah, I was asked to talk about our results in terms of protein targets which might be used to design therapeutics against African sleeping sickness, means Trypanosoma brucei infection. I just went to the laser pointer. Yeah. Indeed, we did a lot of work on that. It all started in 2010, where we had the first observations. But before we go to that, I just want to introduce you with a few words into the human African trypanosomiasis. I'm not an expert of that at all, but my job is to elucidate structures of associated proteins. But what we should know, it's a protozoan parasite which transmits the disease. It's transmitted by tsetse flies, sorry. So we have infection of the blood. It could evade the lymphatic system and then the brain finally. And this is also the stage where it can be lethal. It's usually fatal without treatment. There are two major parasites which still affect sub-Saharan Africa, as you can see. This is a graph from 2017. So most cases are related to the Trypanosoma brucei gambiense. The brucei rhodensiense is just the minor course at the moment. We focused on Trypanosoma brucei, which is another subspecies, but which is closely related to both of them. So what did we do? As I said, in 2010, the entire story started, and I was in collaboration with Michael Dosenko from the University of Tübingen, so he's a quite good expert of trypanosoma infections, and he asked me, together with Christian Betzel from the University of Hamburg, if we could help him solving a structure of a protein from Trypanosoma brucei, and this was the cadapsin B. And we, of course, agreed, because we are structural biologists, so we told him to produce the protein in insect cells, because it's a glycosylated protein. He wanted to have the post-translational modifications on that, and he started to do that. We used the gene of the inactive pre-pro cadapsin B, so which contains the propeptide for blocking the enzymatic activity within the cell, and also the signal sequence for ER entry and later on for secretion. And he started to produce this protein. Unfortunately, he was not successful to produce soluble protein. Indeed, what he observed was this one. Sorry, I just have to go back. This one. So if you now focus on this cell, you can see over time that the cell is enlarged into the right direction, and after a specific time point, you can see the onset of a needle-shaped structure which grows and grows. And what you might also see is that there's one end where a lot of action takes place, and it seems that the structure is shifted to the right out of the cell. Of course, this was not what we expect to see if we want to produce a soluble protein in insect cells, but in fact, it looked like a quite ordered structure. So we were curious to see what it is in fact. So the cell was still alive, the structure did not kill the cell, and therefore we could do some analysis. So we did some electron microscopy, as you can see. And indeed, some quite regular objects were visible, sticking outside from the cell. And when we did some slices of the cell, then we could identify almost a crystal lattice, and this is surrounded by a membrane. So it seems to be that we have grown a crystal within a cell, which we did not expect, of course. So we tried to use these crystals, and we first tried to use these crystals in 2010 at synchrotron source. Of course, that time the intensity of the available X-rays was not that intense as today, so we could not use them for quite tiny crystals. But we came in contact with Henry Chapman, who went to Hamburg that time. And together we went to the LCLS in Stanford, we just heard about, to the free electron laser. And we thought that we could use these small crystals, which have an average size of 1 by 1 by 11 micrometers, roughly, so that we can use them as targets for his newly developed serial femtosecond crystallography approach. For that, of course, we had to isolate the crystals out of these insect cells, and luckily this was possible, so we could just lyse the cells. We figured out that these crystals were quite stable. So this is a scanning electron micrograph here, and you can see even in the vacuum, the crystal was quite stable, which was surprising for a protein crystal. And we could use a slurry of almost pure crystals then that we injected by liquid jet techniques into the pulsed laser beam of the LCLS, that's a CXI beamline. And so we collected single pulse diffraction patterns if we hit a crystal. If not, we had an empty pattern, and we can use that, could use them. Finally, by the way, this is one of our most prominent single pattern diffraction our diffraction patterns. You can see it forms almost an 8, which was quite nice. So after summing up all these single pulse diffraction patterns, we finally had a complete data set, roughly 360,000 frames with Bragg spots were obtained. Unfortunately, this was only corresponding to a hit rate of 9%, so most of the crystals went through the beam without being hit. This was a bit wasting of sample, but in this case, we had enough, so this was not a problem. From that, we obtained 156,000 index patterns, and finally, we could solve the structure of this pre-pro catapsin B from Trypanosoma brucei at 2.1 angstroms. And the good thing was that indeed we could observe the propeptide, which was still bound in the active center of this cysteine protease, so blocking the enzymatic activity. And we could compare that with that of the human pre-procatepsine and could figure out differences which might help in designing specific drugs following the same binding strategy than the propeptide, which is of course the native inhibitor of this enzyme. So the cell provided us with crystals. Our job was quite easy. We just had to isolate the crystals out of the cell. And then nature also provided us with basic information about inhibitor binding to this Trypanosoma brucei, catepsin B, the cysteine protease. But it's not our job to design drugs, so we just provided the structural information, and now others have to work with that. Anyway, we were just inspired by this, what we call now in-cellular crystallization, means the formation of crystals from recombinant proteins in host cells. And we tried to further work on that. Before I give you some more information about that, I have to mention that not only XFELs are suitable to collect serial diffraction data. After we solved the structure of catepsin B, we tested if we also could use these crystals for serial diffraction data collection at the Petra 3 synchrotron source in Hamburg at the DESI. So together with Glaparenko and Cornelis Gatti that time, we loaded our slurry of crystals within a standard nylon loop, which is used for single crystals for the rotation data diffraction usually. And we diffracted this slurry in a serial helical line scan approach. That means we moved the nylon loop in a helical way. So we had 120 parallel helical scans, which have been spaced 5 micrometers apart. exposed roughly for 1 second, which was quite much that time, depositing quite good dose. But we were able to collect sufficient diffraction data finally from less than 100 individual crystals. Of course, we had this needle shape of the crystals, and so it could happen that we hit one crystal several times, producing several diffraction patterns. And this enabled us also to solve the structure of the pre-pro cadepsin B from Trypanosoma brucei. Now, of course, at lower resolution. Of course, we don't have the advantage of the diffraction before destruction, which is, of course, valid for the SFX method at an XFEL. But anyway, if we compare the bioinformation obtained by SFX at the XFEL and by serial synchrotron crystallography at P14, it's almost the same bioinformation. That means, depending on the available d-beam time, you can perform these serial diffraction experiments on both sources, of course, depending on the diffractive volume of your crystals and of the detail of bioinformation that you want to see. So I told you the results of cadepsin B inspired us. So we set up a kind of pipeline, which we call InsectCrys pipeline nowadays, to systematically screen different recombinant proteins in living insect cells for spontaneous crystallization. And in brief, it works quite easy. So we just amplify our gene of interest. Then we clone it in a so-called baculovirus transfer vector. We transform E. coli cells containing a baculovirus genome, and we use the recombinant baculovirus for a gene transfer into the insect cells by transfection. Then our insect cell produces the virus, we amplify the virus, and then we infect finally cells to screen for crystallization. And during the past years, we figured out that we can use the cellular compartment as a suitable screening method. So every cellular compartment has different chemical environments, as you know from standard conventional crystallography. There you change buffer, pH, and whatever. We just changed the localization of the proteins in the cells. So we fused signal sequences for peroxisome at the C-terminus, for the secretory pathway at the N-terminus, for the ER, for example, at the N and the C-terminus, and so on. And we also have the modification for HA and his tags. So if the crystallization fails, we can just isolate the soluble protein and try to crystallize it by conventional methods. If we have a crystal in a cell, we don't isolate the crystals anymore. We just use the entire cell containing a crystal, go to a beamline, XFEL, or synchrotron source, and diffract the crystals in the cells. And this worked without too much background, and we could solve structures out of that. So this is what we established within the past 10 years, to be honest. And some good results are now in the focus. First of all, I want to introduce you to our star. This is the luciferase. You can see this is the poor insect cell, which produced a large crystal, so roughly up to 180 nanometers in length. You can see that the membrane still surrounds the crystal structure, so the cell is not dead, it's still alive, which is amazing. Cadepsin B, I just introduced you to. IMPDH will be the next target from Trypanosoma brucei, also forms large needles, but we can also crystallize fusion proteins. This is GFP fused to an reoviral protein from avian reoviruses, and since GFP has green fluorescence, we could easily follow the crystallization process. But what is really good that the compartment screening works. So proteins have a preferred compartment where they like to crystallize, cadepsin B in the endoplasmic reticulum, which makes sense. Of course, this is the native pathway the protein traveled through in the trypanosoma. And it's also glycosylated, by the way. Therefore, it makes sense that it likes to crystallize there. IMPDH and the peroxisome, this is also the native compartment where we find the protein in the trypanosoma. GFP-MUNS crystallizes only in the cytosol, luciferase also in the peroxisome. So it means indeed the compartment screening is a good approach to test the crystallization behavior of different proteins. And this, over the years, resulted in a lot of interesting, almost crystalline structures. You can see just this gallery. A lot of needles from viral proteins, from human proteins, from fungi, from trypanosoma and so on. So there is no species limitation, there is no kingdom limitation. It just depends on the individual protein if it forms crystals. Unfortunately, not all proteins form crystals in sufficient quantity that we can use them to collect data sets. So this is still a limitation on which we are working at the moment. However, the small stars or asterisks just show that these structures have not been solved from proteins by conventional crystallography. So these proteins have not been crystallized so far using the standard approaches. Maybe this cell has a trick to do it that we don't know at the moment. So it's a kind of complementary approach, how we can call it in this way. Now I will focus on the second protein from Trypanosoma brucei, and this is the inosine monophosphate dihydrogenase. This was crystallized in Hi5 insect cells. I also have some videos for you. So when we follow this cell, for example, you can easily see that once again, a needle appears over time. You can see the hours after infection with the baculovirus. Means this is 1 day after infection, and then we focus on a single cell because the polyhedrin promoter that we use starts to be active 24 hours after the cell infection. But you can clearly see the cell is still alive and the needle-shaped structure is formed out of that. What we also observe is that the structures can move through the cells. They are not fixed. Sometimes they break, but this does not inhibit the growth of the crystal. So the crystal is still stable afterwards and the cell is not dead. The membrane is still around, which is also quite amazing. And what we also can observe is in this video, can see once again the crystal grows, then it's bent, and the bent crystal still continues to grow. So cells can do incredible things with proteins in terms of crystallization, I have to admit. Anyway, of course, since we were also successful to crystallize the IMPDH also in our insect cells, we tried to solve the structure. Before, we wanted to verify that we indeed have a crystalline state of the IMPDH, and one of the most prominent methods to do that is the transmission electron microscopy. Of course, it has the ability to clearly resolve the crystal lattice within the structures. So you might see here within the slice of the cell the quite well-defined and very sharp edges of the structures. And if we zoom in, increasing the resolution, then finally we can indeed see the crystal lattice. So we should expect the fraction of X-rays using these structures. That time we were interested in IMPDH. Of course, IMPDH is involved in the purine salvage and interconversion pathway of Trypanosoma brucei. That means inosine monophosphate is converted to ATP or to GMP and then to GTP, which are the nucleotides important for cell division and so on. So if you use enzymes involved in this pathway as potential drug targets, then you might kill the parasite within the human bloodstream if you, of course, exploit the differences between the human and the trypanosoma enzymes. And therefore, this IMPDH was in our focus. Indeed, we were able to isolate the crystals once again from the cells. So this was also done a few years ago, and therefore we isolated the crystals out of the cells. We went once again to the CXI beamline at LCLS, to the XFEL, and performed a method of SFX with 40 femtosecond X-ray pulses. This time we had a hit rate of 3%, which is quite low. But roughly 30,000 crystal hits were sufficient to finally solve the structure once again at 2.8 angstroms. So that also shows you that cells are able to crystallize the proteins in a quite pure way, despite the crowded environment in the cell. So the protein is surrounded by thousands of other proteins, but crystallization, which can also be a purification step, as you know, works well to form crystals that diffract to high resolution. So this is also quite interesting to see. The next highlight here is that we were able not only to solve the APO enzyme, we found regulatory nucleotides bound to so-called canonical binding sites, which have been described before. So we found a dimeric structure within building up the crystal within the asymmetric unit. And this contains every monomer binds an ATP and a GMP at the described canonical binding sites. And this has been in discussion before, which nucleotides can bind to the IMPDH. Usually what you do is you crystallize the protein, you soak the protein crystal with high concentration of ATP, and then you analyze for ATP binding, or you soak with high concentrations of GTP and ATP together, and you have a look for the competition. Here, all different nucleotides have been present because the crystallization occurred within the cell, and therefore the highest binding, the binder with the highest affinity won the race and showed us the structure within the binding site. So we could identify, hopefully, the correct native cofactors which bind to the binding sites. And this is also quite an advantage of crystallizing proteins within a cell. This structure was also confirmed by series synchrotron crystallography, once again at the P14 beamline at Petra 3 synchrotron source in Hamburg. What we did now was we measured the crystals within the living cells. So we loaded the cells on a micro mesh mount. You can see the individual cells, and then we once again raster scanned in a helical way this mesh mount. You can see that we, at some position, we obtained diffraction patterns from individual crystals. We had a beam size of 5 by 10 square microns, less exposure than during the first experiments because the intensity of the source was much higher now. But finally, we were able to solve the structure once again, of course, at slightly lower resolution, as we discussed before. But here, just 8,500 hits were sufficient. We had a better hit rate. Of course, we had a higher crystal density on the mesh. And finally, roughly 5,000 crystals enabled us to solve this structure, which was super impossible to that that was solved before by SFX. So for IMPDH, I told you it's important to have a look at this canonical nucleotide binding sites. We could clearly reveal that we have ATP bound to the first canonical binding site and GMP to the second canonical binding site. Nowadays, we know from our series synchrotron crystallography within the living cells that this is not GMP, it's GDP. And one phosphate was hydrolyzed during the isolation process of the crystals from the cells, which is an argument why crystals should stay in the cells and diffraction data collection should occur using the living crystal containing cells. Then you avoid these hydrolyzation effects and this purification effects. So these nucleotides are suggested to be involved in the regulation of the enzyme activity, because this is the so-called regulatory Bateman domain, where the two monomers are linked together to form the dimer. And depending on the presence of the nucleotides, an octameric state, which is formed by 4 of these dimers, has a more open or more closed central core. And the more open central core is associated with the active state of the monomer, of the IMPDH, sorry. And this is just regulated by the angle in which these dimers interact with each other, which is impacted by the nucleotide binding. That means what we found is the inactive state, which nicely corresponds to further investigations. So if guanosine nucleotide is bound, a guanine nucleotide is bound to the second canonical binding site, then it's always the inactive form. If this is exchanged against ATP, then we have the active form with the larger channel in the middle of the octamer. But now we consider that this combination, if this is GDP, this is the native combination, not influenced by any artificial concentrations. And finally, we tested a third enzyme, which is the GMPR, which is the guanosine 5' monophosphate reductase, which catalyzes the reverse reaction from GMP back to IMP. Once again, we could obtain structures within the cells after baculovirus infection. We also went to a synchrotron source so far and performed the series synchrotron crystallography, once again at Petra 3 in Hamburg. And I hope you can convince you that you can see diffraction spots here up to roughly 3 angstrom resolution. So we are currently on the way to process the data. The data quality is not that good as we have obtained for IMPDH. And therefore, we do not have a structure so far, but we hope also to identify the native cofactors. Of course, this enzyme is also regulated by nucleotide binding. So maybe also the cellular environment helps us to identify the right cofactors. So what I told you within this brief talk is that cells can, of course, crystalline proteins, and these in cellular crystals, this is how we call them, can be used as targets for structure biology. You can perform serial femtosecond crystallography at an SFX if you have access, which is the major problem nowadays. Or you can go to a synchrotron source and perform serial synchrotron crystallography, of course, depending on the diffractive volume, means on the size of the obtained crystals. This is not a rare event. I showed you a lot of examples, and we also solved other structures than I introduced to you today. Can be dynamic process, so sometimes crystals grow and shrink and grow and shrink. And the good news is, if you have the recombinant baculovirus and you infect your cells, you will know 2 to 4 days after infection or transfection if a crystal appears or not. So it's a quite quick process. Stability varies on the protein. Some crystals can be isolated, some not. But as I told you, nowadays we prefer to collect the diffraction data directly from the crystals in the living cells. And using the example of IMPDH, I try to convince you that the intracellular environment provides all the cofactors. So the method has the potential to identify the right cofactors, the allosteric cofactors for enzyme activity regulation. Our problems are that we still do not know what's the contribution of the cell to the crystallization process. If the crystals are quite small, nanocrystals, we have a problem to detect them. And the crystallization efficiency is frequently quite low. That means we observe regular structures, but we do not obtain sufficient structures to perform serial X-ray diffraction data collection. That's it, what I wanted to tell you. Take-home message is not only for enzymes involved in infectious diseases, for example, in Trypanosoma brucei infections, also for all others, try in-cellular crystallization as a complementary approach. You might fail, the probability is high, but you can obtain lucky hits, and sometimes these lucky hits have not been obtained before by conventional methods, so it could be a complementary approach indeed to be tested. If you need further information, please contact me. I'm always happy to provide our knowledge and to spread our knowledge and to help if you want to test it. I have to thank a lot of collaboration partners who have been supporting my group during the past 12 years in developing all this stuff, and of course the beamlines and the funding agencies. And finally, all people who have been involved from my group. This is also not an actual picture, but Mia Lai-Rudolph, Johanna Boger, and Robert Schön are the key players of the data that I presented this afternoon. Yeah, thanks for listening.
Very nice talk. Thank you very much, Lars, again. Questions, anyone in the chat, or you can put your hand up as well. Okay, just a moment. Right, so those are questions. So I'll ask, oh, here we go. So there's one from Lawrence, go ahead.
Yeah, so I was wondering if, what's the role of the cell membrane in the crystallization process? And could you make sort of, could you culture cells to make the liquors for in vitro crystallization? So the idea is like, so in the lab, we're crystallizing proteins, trying to find the right, physiochemical environment.
Yes.
But it seems like the cell provides that. And if we can make a, I guess, a artificial cell or a membraneless cell, I guess is what I'm trying to make, or a cytoplasm in a glass, I guess is lack of a better word. But that's probably cultivating a lot of cells. So is that worth it? What do you think?
Well, as I mentioned, we don't know the real contribution of the cell. What is almost clear is that the same principles for protein crystallization apply also in the cell as we know from outside the cell. So we need a supersaturation, we need a very high concentration of the protein, and we need specific chemical environments. So membrane impact is, in my opinion, just that we have a kind of compartment where the protein is transported to, and this helps to oversaturate or supersaturate the protein solution to reach the highest concentration that we need in the specific compartment. We don't have any indications that the membranes are involved by specific interaction or whatever.
I see. Okay. Thank you.
So in designing artificial compartments for crystallization, I think just use a cell. It's the most easy way, more easy than to design membrane vesicles or whatever. Because if it's true that it's only a concentrating effect, yeah, then you can just saturate the solution in small volumes, and that should have the same effect.
Thank you. Thank you.
Any other questions? Okay, I don't see any other questions for now. So why don't we just thank our speaker in the interest of time. Thank you very much again, Lars. Wonderful talk. And of course, you can always field questions through the chat or directly to him.
Of course.
At a later date. So that brings us to the end of our plenary session. We had a wonderful time, lots of very interesting talks, and hopefully lots of insight gained from those talks. We shall now go to a short break. I think it will have to be very short, as the other breakout sessions will start very soon, and I'll hand back over to the organizers. So thank you again, everyone.
All right, so we're going to take a short break, and we have to create the breakout rooms in Zoom. So we're having two parallel sessions. I'm going to create the breakout rooms. One will be for the mineralogy talks, and the other for the earth sciences talks. I don't know if Zoom can create the breakout rooms on the fly. So if I do this and it somehow stops the Zoom meeting, please just log back in. Making breakout rooms on the fly might screw the pooch, as they say.
Hi, Lawrence.
Yep.
Can you just start with a Brian for us?
I didn't hear you. What did you say?
Brian, can you try Brian for us, because that's the way it was created yesterday.
I actually did not. I can barely hear you.
Yeah, what I'm saying that can you just start with Brian for them to do because I'm trying to read, because they did it yesterday for us.
So you're saying have a chat with Brian, yeah.
Yeah, for the breakup room.
Right.
Yeah.
Brian Massara. Okay.
Yeah, Brian, yeah.
All right. I will do that.
Okay.
Let me. Okay. Well, let's take a break while I do this.
How long do you want to allow for the break?
What is it? 10:34 now. Why don't we take 10? So we will be 15 minutes behind. Is that fine?
That sounds good. That sounds good. Yeah.
Are the speakers here? With apologies to the speakers who are 15 minutes. We're 15 minutes behind schedule. Okay, thanks. So I created the 2 breakout rooms and it didn't seem to break anything. So if you want to go to a breakout room, just look at the very bottom, there's a little button that says breakout rooms. And then if you select that, there's 2 options. One says mineralogy and one says earth sciences. And that's the topics of the 2 sessions that are coming up. So the people who are left in the unassigned room, so this was the room that, let's call it the main auditorium. There's 27 people. Are you able to see breakout rooms button down at the bottom? There should be like looks like a Microsoft, I don't know, little 4 square thing. And there's a red number.
Yes, I do. I just arrived, so I'm going to move to. I need to check the program. Okay.
Okay. And you can use the same button to go in between the two rooms if you can do that at any time. You can so if there's talks in both sessions that you want to see, you can. It's just like a normal conference. You just sort of get up and go to a different room. Hello. So in this room actually will be not active. So if you can, please go to one of the breakout rooms. Actually, we have to close this room, which means that you're going to be assigned to one room or the other. So I can give you a minute to pick a room yourself. If not, I think the system is going to randomly assign you to a session.